Thank you so much, Steffi, and a huge thanks for this amazing, amazing meeting. I've never been at a meeting like this before, and I think you set new standards for how we should conduct our research and certainly the participation of all the patients. I am completely honored to be introducing one of my great friends. So Professor Catherine Briskin obtained her MD and her PhD in biophysics from the University of Göttingen, Germany. This was followed by postdoctoral studies at Whitehead Institute, MIT, where she started her work dissecting in vivo role of reproductive hormones in the mammary gland. She's held academic appointments at the Cancer Center for Mass Gen Hospital at Harvard Medical School and at the Swiss Institute for Experimental Cancer Research in Lucerne. She then subsequently joined the Swiss Federal Institute of Technology in Lucerne and also holds a position at the Institute of Cancer Research UK, where she is a team lead. Catherine is an exceptional mentor. She has served as Dean of the Doctoral School and holds several prestigious EU training grants. She also serves in the she also serves on the AACR Women in Cancer Council. Her research focuses on cellular and molecular functions of estrogen, progesterone, and androgen receptor signaling in breast development and cancer. Her pioneering work on the development of the introductial mind model has had an enormous impact on the field and has changed the way that we all conduct our in vivo steroid signaling studies. So this morning, she is here to talk to us about her work on lobular carcinoma development via the teat. Thank you, Catherine.
It's on now. So can everybody hear me? Yes, that's good. Okay. So I thank Leonie for the flattering introduction and the lovely organizers for putting together this very stimulating conference. And I think, among us scientists, there was a consensus that this was really impressive, what we listened to the first days. And the advocacy movement stipulated by Stephanie, I think, has really made things move and made enormous progress. So congratulations to all of you and keep going. So I'm putting up a slide here that is about 10 years old to point to what has been a big problem in the oncological field, and that despite the enormous progress we have made in cellular and molecular understanding of biological processes and cancer, many of the drugs that we have proposed coming out of the lab actually failed as they were translated into the clinics. And that has had a lot to do with the way the models we used. So it's understandably you want to use a model that is practical, that gives you a result quickly, and that doesn't necessarily mean that that model reflects what goes on in a patient. And so that was particularly the situation was particularly dire, if you wish, with regards to estrogen receptor positive breast cancer, that, as we all know, are the majority of breast cancers. And that has to do with some sort of surprising findings, and that's that as much as we manipulate the genetics of the mouse, the tumors that grow out in mice are typically estrogen receptor negative. And we still don't quite understand why that is so. And then we can also xenograft breast cancer cell lines. And again, there it's surprising that although most breast cancer are estrogen receptor positive, only a minority of breast cancer cell lines are. And then when you graft them into mice, a minor fraction grows there. And only so if you supplement the mouse with exogenous estrogens. And that creates artifacts because most breast cancers occur in postmenopausal women who have very low levels of estrogen. And we know that estrogen levels will change dramatically how cells signal. Then it's been very difficult to actually get ER-positive breast cancers to grow in the subcutaneous tissue. And with a revolution sort of stipulated by work spearheaded by the Clevers lab, we've become very good at growing organoids from individual cells. But here again, it was not so successful in generating such organoids from estrogen receptor positive breast cancers. And so I think we can congratulate all of us as a field that we've made progress in recent years in increasing the repertoire of models that are now available for this very difficult to study lobular cancer. And here is the hero in my lab who is in love with lobular cancer, George Flommers, who's put together this review and made this nice scheme. And you see, so many people here in the audience who have contributed to increasing the repertoire of tools that we have now available. And we also saw a beautiful example of lobular cells growing in zebrafish yesterday from our friend from Edinburgh. So I will be talking now on sort of our contribution, which is here with the xenografts derived from cell lines and from patient ILC cells. And what really made the difference has been about the where do you place your tumor cells. And so as a field, we have been injecting tumor cells under the skin, and that poses a big challenge because that's not where tumor cells come from. So it's asking them to grow in a metastatic site. And so Fariba Bebot in Dan Medina's lab had started injecting cells via the teat, that's what we call the nipple in mice, to place cells inside the milk ducts because she wanted to study in situ disease. And that's what we tried as well to see what happens with the ER-positive cells. And we modified that by introducing a lentivirus that would allow us to trace the cells with a GFP and with luciferase. And so once you have injected the cells, you can monitor them in the different glands in vivo by bioluminescence. And when you open the mice, you can find your green cells. And so we were able, with one cell line, the MCF7 cells, which we get to grow in the fat pad in the presence of high levels of estrogen, to compare to what happens when MCF7 cells grow inside the milk ducts. And this revealed that it really has a major impact. More than 2,000 genes were differentially expressed. And it revealed that the cells injected into the subcutaneous fat are exposed to TGF-beta, and as a result induce a basal differentiation program. So they start expressing P63, vimentin, and markers that have nothing to do with luminal epithelial cells. Whereas by putting them into the intraductal environment, they retain their luminal properties. They upregulate estrogen receptor, androgen receptor, growth hormone receptor expression, and maintain their luminal phenotype. So, having shown that this microenvironment is so important for the biology of the cells, we went on to set up a pipeline where we would get patient samples, dissociate them in the lab. And I mean, we get very small samples from tumor ectomy surgeries. Dissociate them to single cells that are infected. And so we put these green and luciferase expressing cells into mice and then watched what happened over time. And in the end, analyzed these engrafted glands. And so we've been doing that for years. It takes very long. And there have been several lessons learned. And so that as we put these tumors out, T cells come all from invasive tumors. In the mice, they are reverted to an earlier stage. Not so many become invasive actually in the mice, but they recapitulate earlier stages. The tumor subtype is retained, indicating that That's a tumor cell intrinsic feature. And the proliferative and metastatic propensity of patient tumors are also cell intrinsic and maintained in the PDXs. And we observed that the response to endocrine therapy is also preserved in tumor cell intrinsic. And so we think they're useful tools and remain prognostic and predictive. So these times are very exciting because now we can address the question that has been bothering us. How is it that estrogen, progesterone, and androgens actually impinge on this process of ER-positive breast carcinogenesis? So now we do have tools to study that. Yeah, we had to learn that we have to be very patient because it's a very lengthy process. And we have learned some lessons also about metastasis, yeah, where we thought, now, great, we have spontaneous metastasis in this model, but we were unable to study these metastases because the cells are so sparse. And in fact, a very talented graduate student, Patrick Wauert, showed that the model correctly mimics the process of dormancy, that the cells take on a mesenchymal fate as they reach distant organs. And by switching on an epithelial fate with forced expression of eckaterin, we can get them to wake up and grow into macromets. So here is another lesson we learned. So now that we had a couple of patient-derived xenografts in place, we stimulated them with estrogen and progesterone, or the two hormones combined, to mimic the levels of hormones we find in premenopausal women. And so what you realize, it looks rather messy. So you have all sorts of combination, and each patient's tumor is in a way different in how it responds relatively to estrogen, progesterone, and the two combined. Yeah, when you summarize that, overall, estrogen has a positive effect on proliferation, and progesterone has a more binary effect. It can either stimulate or not have an effect. And the two hormones together have different effects on the tumors. And when we summarize the results from 9 different PDXs, you can see that both estrogen and progesterone stimulate growth of these primary tumors to a similar extent. The two hormones combined do not significantly affect tumor growth. But then when you look at metastatic load, yeah, all 3 conditions promote metastatic load, indicating that the primary tumor growth and the metastatic process are dissociated. So we can have one, it's not just proliferating faster gives you metastases faster, but here are distinct biological processes involved that are differentially regulated by the hormones. So those were the general lessons. Now let me turn to lobular. And as I said, George was very eager to look at lobular in this model. And so he took the 2 classical lobular cell lines, as well as 2 non-ILC ER-positive cell lines, which we saw yesterday also in Nats talk, and engrafted them into mice. And you see that the take rates are slightly superior for the non-lobular breast cancer cell lines. And then the in vivo growth is faster for the non-lobular versus the lobular cell lines shown in the purple color, as you would expect from the clinics. Now, marvelously, yeah, I like to look at the mouse mammary gland. You see the mouse ductal system here in a virgin mouse. The estrogen receptor positive MCF7 cells dilate the ducts, and I call this the sausage pattern, owing to my German origins. And sure enough, we get the Greek grape pattern when you grow lobular cells, and this is amazing. Yeah, so we transcriptionally, there's and so forth, there's not all that much difference between the different models. And here you have a completely different morphogenetic process with these grapes appearing, indicative of the motile properties of these cells. And in fact, they induce morphology within these mouse milk ducts. Yeah, you have beautiful histology, comparable to what Stuart and others showed us yesterday. That's quite nicely recapitulated. And George detected metastases, analyzing mice. Before 3 months, there's not much to be detected. But then after a prolonged period, and here going up to a year, you see that there is metastatic load in various organs that reflect what is clinically relevant in the clinic. And so George has now characterized the 134, the SUM44, and more recently also the IPH926 cells, which are triple negative, generated by Matthias Kriskren from Hanover, and shown that they all recapitulate the whole process of lobular carcinogenesis. Now, having established this, he went on to ask, now that we have this system where we are able, and here I'm showing you actually primary tumors from patients that engrafted as PDXs. And some of them and they are very slow growing. I keep on stressing that. So George managed to get enough expansion over 2 or 3 generations of 3 of the ILC models and 3 NST models to allow him to isolate cells, which he did by FACS sorting based on the GFP, and profile them by RNA sequencing. And while I was skeptical about such a small experiment, yeah, the comparison showed that CDH1 targets were down in the lobulars, and there was the signature of lobular versus ductal. So we gained confidence in trusting the outcome of this experiment. And as he did further bioinformatic analysis, you see different databases used here, and always at the top speaks to the extracellular matrix, collagen formation, ECM regulators. So next, he asked, well, what we find now in this model, is that relevant to patient samples? And took the matrisome signature that Richard Hines' lab has developed to look at TCGA database. And you see it's far from clear, yeah, but you see an enrichment of red color on this part of the graph, where you also see an enrichment of purple lines that reflect the ILC. So it seems that the ILC tumors tend to have an increase in ECM signature. Now, how can that pan out for something practical? It's difficult to interfere with the ECM, but there are enzymes that modify the ECM. And as George and Renee compared lobular versus non-lobular ER-positive tumors, here you see that CDH1 is a top differentially expressed gene, and then they looked at the whole family of the LOXs, and LOXL1 and LOXL2, 2 family members were so this was, LOXL1 was increased in the lobulars, as was elastin, and also collagen 1A was increased in the ILCs. And so that gave rise to the idea that here's an enzymatic activity, and LOX is in fact a secreted enzyme that cross-links collagen and elastin. So we had both the substrate and the enzyme increased, and that led us to try and see whether inhibition of LOX could slow down lobular tumor growth. And this can readily be done with a natural compound called BAPN that's isolated from this plant. And very importantly, what it's actually took us by surprise, we had isolated purely the human tumor cells. And yet we had found all these extracellular matrix genes. Yeah, when I had been taught those collagens and everything is made by the matrix, by the fibroblasts, and myofibroblasts will upregulate LOXs. And so I thought that was very surprising that in fact we find these genes expressed in the carcinoma cells. And George checked with RNAscope for where LOXL1 is expressed. There is some expression in the stroma, but then, you know, this is looking at patient samples. But then here, for instance, you have an example where it's clearly and most exclusively expressed in the ER-positive tumor cells. So really that is a very unexpected lobular-specific feature that these extracellular matrix proteins get made by the tumor cells. Okay, so here we go with the BAPN treatment. First, George tested in the early setting. So 3 weeks. 3 weeks after injection of the lobular cells. He put them on BAPN in the drinking water. I click on this, but it doesn't. Okay, well, anyways, you can see it, right? So, it's okay. No, thank you. Sorry. So here the cells were injected for 3 weeks, and a 3 months treatment starts. And you can see that the vehicle-treated cells grow faster than the one exposed to BAPN. And at the end of the experiment, when the tumors are stained for serious red, which reveals fibrillar collagen, you see a difference in nice linear fibers, and they are more disrupted in the BAPN exposed glands. Now, in a metastatic setting where the tumor cells have been left in mice for 5 months, and then for following that is a 3 months treatment with BAPN, in both the MM134 and the SUM44 model in various organs, there is a significant decrease in burden of metastatic cells. And as he took this on to try on PDXs, there are 2 examples here. There is a trend to respond in this particular lobular patient sample, and a significant response in this other lobular patient sample. And there was not a response in the 2 non-lobular patient samples. So in the SUM44 model, George analyzed the expression profile, and it looks like to me as if that was of a chemotherapy. So you found a lot of proliferation-related genes down-modulated, and interestingly, it also had an effect on estrogen receptor signaling and MYC targets. So the current working model is that the LOXs by cross-linking elastins and collagen enable this growth that is so critical for the lobular carcinomas, and that we, by inhibiting LOXL1, we can disrupt this, and this will lead to down modulation of ER signaling and MYC targets, and reduce cell proliferation, invasion, and metastasis. Now, the problem with this so far is that the BAPN is not tolerated by patients in the clinics. And this lady here, Renée Flaherty, is postdoc in London. She dug out a company in Australia that's making LOX inhibitors. And so she found these compounds and tested them and could show that they are better tolerated by the mice, but, as assessed by weight change than the traditional BAPN, and started setting up some experiments. And simultaneously, she improved on our ways of measuring and quantifying these collagen disruptions by using a software developed from Eric Sahai's lab. And so you can see here by different parameters, you've seen changes induced by this drug after a 2-week treatment. And interestingly, as she looked at the endogenous organs, there were no significant changes apart from the BAPN treatment. So having shown that, yes, you went on to show then with the 134 series that there is a decrease, and you can sort of be a bit skeptical about this sort of IVIS curves. But it has to be said also that the mouse facility was in crisis over that time. Anyhow, at the end point, you can clearly see a difference in the Ki-67 and differences in the collagen structure. And it looked similar with the SUM44. And this stimulated Pharmaxis to provide Renee with the drug in the chow. And that enabled her to perform more longer-term experiments. So she set up the SUM and the 134 and also the IPH model to do sort of proper treatments with where she would have just the vehicle chow, and to mimic the clinical situation of aromatase inhibition, do an ovarectomy, and then have those two conditions with the drug. And only in the case of the IPH 296, which are triple negative and which she showed to be unresponsive to ovarectomy, she just did the control and drug treatment. And so, mind you, so the mice have been bearing the 134 cells for 5 months. Then they undergo ovarectomy, and then the drug treatment starts. And here you're just seeing what the weight change, and as expected, when you overectomize mice, their weight goes up. Yeah, but it's also an indication that otherwise the drug is well tolerated. And so here is an overall picture. So until day 150, the different cohorts are growing, and then the drug treatment starts. And when you analyze that, you see that there is an effect of a significant effect when you compare the vehicle versus ovarectomy and drug. And the drug also adds the drug together with the ovarectomy is more efficient than the ovarectomy only in this model. And looking at lung metastasis, there's also significant differences with the addition of they respond to ovarectomy and they respond to ovarectomy plus the drug. She then analyzed the 134 xenografts, and here you see that so this is, still the 134, now looking at the histological examination. And so you see that Ki-67 is decreased by ovarectomy and by the drug, by the LOX inhibitor, and the two together decrease it further. And similarly, then there are these changes that occur to the collagens. Now, the second model, the SUM44, responds a bit different in that the effect of the drug is sort of as good as is ovarectomy, as is mimicking the AI treatment. And yeah, so ovarectomy and the LOX inhibitor treatment both bring down tumor growth and don't have a synergistic effect in this model. And then at the histological level, looking at Ki-67 index, again, you see ovarectomy and drug bring down the proliferative index, and the combination, however, doesn't add to that. So finally, in the triple negative model, you see again an effect of the LOX inhibitor with a decrease at the end point of the total flux. That is, there's a trend. It is significant when looking at the lung metastases. You can see the differences by the IVIS and in the histological sections. And it's kind of tricky with the in vivo experiments, and oftentimes the readouts by histology are more consistent, as you can see here again, with the Ki-67 index being significantly decreased. So I conclude saying that it looks hopeful for these LOX inhibitors. We still need to figure out how much that plays into matrix stiffness, how consistent will be the effects of estrogen receptor signaling. But it has an effect apparently on tumor growth, invasion, and metastasis. And yeah, we expect to find more answers by analyzing the omics from the tissues that were collected. I want to perform targeted genetic screens to better understand what are the genes cooperating with this, and then try and set up a trial with colleagues at the Royal Marsden in terms of a window trial where, yeah, the challenges are to have a, so the good thing is the drug is well tolerated and Pharmaxis is happy to provide it. But we need to carefully think about the inclusion criteria, and the Ki-67 index that is nice in the models is likely going to be lower in the patient and might be more difficult. So we will target also with the collagen content and the omics, and then have to see what will be the best endpoint out of those, and determine biomarkers for the response. In other words, is it dependent on LOX expression, presence of collagens, other matrix proteins, and or the activity of LOX in the bloodstream. And so with that, I'd like to thank all the people involved in this work. In particular, Flavia and Bay helped Renee with these huge animal studies. And George was assisted by Laura in doing those in Lausanne, and then the funding sources. And I thank you very much for your attention.
Catherine, that was amazing. We have a few minutes for some questions. While people are gathering their thoughts, I might ask you one. It was fascinating to see the upregulation in the ECM proteins in lobular versus regular nondescript. We see that when we look in our metastatic tumors, this massive increase in ECM proteins. Do you think if you force expressed LOXL1 in regular tumor cells that you would drive them towards a more lobular phenotype?
You might do so. We haven't actually tried. And would they become more metastatic also,
yes.
Right? Yeah. I mean, actually, LOXL1 is part of the EMT signature also, we noticed. And when you down-modulate ECadherin, LOXL1 goes up. With the results with this pan-LOX drug, yeah, I'm not so sure how much is sort of LOXL1 specific and how much the other family members play into it. Yeah, we started, of course, looking at the others as well. And then you get into that where, yeah, different models have different amounts of these. So I think there is a lot to be seen.
Great. Okay. Here, I'll start here.
Catherine, that was great. The metastasis that comes from them growing in the duct. So how does that work? Do they eventually break open they eventually burst the duct and then they metastasize, or do they? Because I remember when Fariba first did this, it was very controversial as to whether they invade a classic metastasis by invading out of that duct or whether it just bursts open. Do you have a sense? Because for the lobulars, the spread is quite interesting in the peritoneum and all these interesting sites. It would be nice to know that.
Yeah. No, so we've been learning a lot by simply studying the model.
Yeah.
And clearly my first thinking was, oh, wow, now we can study invasion and we look at metastasis. To our surprise, when putting in defined invasive tumors, we have only a third of them after a long time showed any evidence of invasion. Okay. But yet in many of them, we detected cells in different parts of the body. So it looks to me that, yeah, you can have a transient disruption of the basal lamina and have these cells get out and seed. And probably that what happens with the ER-positive breast cancers. Yeah. But what is reassuring, and I mean, I think that sounds gruesome, imagining that there are all these cells sitting in different parts of the body, is that we have found it incredibly difficult to get them to wake up. So they will sit there and fall asleep, and you can die of many other things before they ever think about waking up. Yeah. Now, I wonder whether the lobular might be a bit different in its mechanisms. They are even longer to ever start invading, but then, yes, they grow differently in the fat pad. And in fact, it's with one of the lobular cell lines that we have had a real metastasis to speak of, in that a whole ovary was full of the 134 cells. And we had a bigger brain metastasis than we would have ever seen with the MCF7 cells. Yeah. So I think, yeah, I mean, there might be different biology there between the two types. Right. But definitely in the triple negatives, however, they grew at the same rate in the distant side as they did in the primary side. Whereas the ER positives at the distant side fell asleep.
It's amazing the observation you have. And I have a question regarding the selection for this window of opportunity trial based on Ki-67. So my question is, as we know that there is a subgroup at the transcriptomic level being named proliferative, do you think it may be a possibility to select the patients according to the proliferation transcriptomic status rather than the Ki-67?
Yeah, absolutely. Yeah. I think we need to look for different ways, and I'm afraid that we still need to screen quite a bit and learn quite a bit more before we have a good strategy.
And another question regarding the role with regard to the progression of LCIS in patients. Do you think that LOXL1 could be a marker of progression in this setting?
That could also be. I mean, George had that observation in that LOXL1 seemed to be highly expressed in the invasive parts. So it could also be upregulated during invasion or be responsible for the invasion. These are just observations at this point. We haven't manipulated or induced the LOXL1 expression in this way. Yeah.
Hi, Catherine. Beautiful work. I'm a bit puzzled. So looking at your beautiful paper and the work you presented today, I have the impression that you're mostly inhibiting proliferation. And you might also be inhibiting a migratory process. And my question is, if it is a migratory process, how do you think this works? Because these lobular cells tend to migrate in an amoeboid fashion and are not that dependent on either collagens or fibronectin to bind. So have you actually looked at cross-linking of collagen, so stiffness of the bundles when you do something with Loxel, and are they dependent on that?
So yeah, we have those various parameters on the collagen fibers, right? You can see the curvature, you can see how hyphenated they are. And there is this sort of, and I'm not too expert on this, but you see going away from the straight, strong collagen fibers to more disrupted and more branched fibers.
Right. So it's the structure. You haven't looked at the actual stiffness of the bundles.
And the actual stiffness, no. So that's not so easy. And yeah, we're currently getting on board by doing NMR elastography. That's sort of the next step. And talking to Valviva to see what we can do. Yes.
Very innovative work. Beautiful. Simple question. Do you have to do survival surgery to take those mice out plus 5 months?
Do we need to do survival
surgery on their primary tumors? Yeah.
No, it's the postdoc that I need to do survival surgery on.
So So the size doesn't limit the end of the experiment, the size of the primary tumor?
It's always the primary tumor. It doesn't get to be such a burden. Yeah, with the MCF7, it's a bit different. Yeah. So we've left them in for a year to get those metastases. And at this primary site, it was still okay. It's different for MCF7. Those after 7 months, you need to intervene.
Okay. I think that's our time is up. Huge thanks to Catherine. Okay, and I'll hand over to the next chair.
It's my honor to be chairing this session. I have to say, I really love this meeting because I get to see in the people whose work inspires us on a daily basis. And it's also neat to be in the presence of the women that have helped to create this community and to sustain it, and that for which all of this work needs to be done. So in that regard, in terms of the inspiring work, I'd like to start by introducing Dr. Adrienne Lee. Dr. Lee was recruited here in Pittsburgh from Baylor University in 2010, where he is now the director of the Institute for Precision Medicine. And he's going to tell us now about his basic science studies on ILC and how they're translatable and the basic mechanisms that are being learned to facilitate translational work.
Okay, welcome. Everyone can hear me? That's great. It's a good start. So I want to thank the organizing committee for giving me the opportunity to speak. Okay, that's good. You guys aren't asleep. That's a good start. I was thinking you weren't going to laugh. And I will be talking about analysis of cell lines. This actually follows on very well from what Catherine was just talking about. I actually gave a talk last year in Utrecht at the same meeting. I actually spoke with George Flomos kind of back to back. We did a talk together on models, and this is actually his slide. If you haven't read, Catherine pointed this out, but if you haven't read it, it's a really amazing paper, what he put together. This atlas of lobular breast cancer models, challenges and strategic direction. It's really an incredible compendium of everything we know about all the models we have for ILC, and I'd recommend reading it. And so this is his slide, and it's just one slide on why we do need to do modeling and what we learn, as such as Catherine said, what we can learn from these models. So obviously, we have a number of patients here in the room. Hopefully, they had the opportunity to donate their tissue for research if they had a biopsy or they had a resection. That tissue is incredibly important. We can study it directly, or we can then grow it in various models. We can grow it as cell lines, such as the MM134 and SUM44 that you just heard. We can grow it as patient-derived organoids, a growing area that grow in 3 dimensions, and many people feel a bit more representative of the human disease. And then we can grow them in mouse models or as mouse models. Every model is incorrect and wrong. That's a fact. The only model that's correct is actually the patient. They all have their pros and cons. but they each have their own utility. And as long as you know the issues with them, then you can hopefully interpret data correctly. And then we can learn about the disease. And nearly every advance in breast cancer, and true for lobular, has come from these models and this type of research. As I said, I'll just be focusing on cell lines. So cell lines have been essential to understanding the cell cycle and chemotherapy use and many targeted therapies. The largest analysis of cell lines, one of the largest analysis, is something called the Cancer Cell Line Encyclopedia that came, of course, CCLE, came from the Broad Institute. They took like 1,000 cell lines, did every assay you could possibly imagine, and made all of the data publicly available. It was an incredible effort by them to accelerate the field. Breast had 62 models in there, so it was very well represented in terms of knowledge from breast cancer. As you can see at the bottom, there's 62 cell lines. However, of course, lobular is unfortunately underrepresented, and there's only 2 ILC cell lines in there, the SUM44 and the MM134. And those end up being the model that everyone uses. And obviously, 2 cell lines doesn't represent the heterogeneity of ILC. So what is the definition of a lobular cell line? Stuart Snit said, okay, pathologists have trouble actually defining the disease itself using pathology. Well, it's even harder to do a cell line because the disease is diagnosed based on looking at the pattern of its growth. And these cell lines growing on plastic don't have a pattern of growth. So this becomes extremely difficult to know what cell lines are ILC. And interestingly, you can then move to the kind of genomics or the underlying molecular deficiencies in these cell lines. And there was an analysis by the Rather Consortium, sequenced a bunch of cell lines and realized that a number of breast cancer cell lines actually have, as you can see here, mutations in E-cadherin down here that look like classic lobular E-cadherin mutations. Yet the cell lines have a number of like annotations not indicating that they're ILC. So a number of them just have no indication of what tumor they came from. And a number of them potentially are mislabeled or have other labels. And it's possible that these were generated before lobular was of interest. And so they are just purely lobular cell lines that are actually misannotated. And in that paper from the Rather Consortium, they called these ILC-like cell lines and said, well, maybe these might be useful for research. But that was essentially the end of it. And they haven't really been studied further. So about 3 years ago, we set off to perform a comprehensive, similar to what the Cancer Cell Line Encyclopedia did. We said we're going to do a comprehensive analysis of all these cell lines using the same assays they did, make all the data publicly available, and this will be integrated with, so you can do integrative analysis. This was spearheaded by, it's truly a lab effort. It took a lot of work and it took a lot of people to do this, but I should say it was spearheaded by Osama Shah, who you heard from yesterday. He just graduated with his PhD, and helped out by Chelsea. I presume both of them are in the audience. We performed optical mapping, optical genome mapping, SNP arrays, whole exome sequencing, methylation arrays, mRNA sequencing, and reverse phase protein arrays. This, we did it in a way that was similar to how the Cancer Cell Line Encyclopedia did it, so it can be readily integrated with their data, which I think is impossible because we didn't study ductal cell lines. This is funded, I should point out, by the NCI through something called the Oncology Models Forum, which is a part of the NCI that's important, that feels this is important, the generation and characterization of models, and then the dissemination of that data. Through that funding, we do this in collaboration with the Cancer Cell Line Encyclopedia, because they were the ones that did it originally. We reached out to them, and so they are doing drug screening and CRISPR screening on all these cell lines as well. And if you go to their portal, some of them have already been completed and you can go and grab that data. So that's all publicly available as well. And we did, when we started this, say we were going to do phenotypic characterization. It turns out every lab does this differently. And I don't think as a gold standard that data is that useful because everyone's going to get different data. I think so we focus mostly on the omics characterization. I would point out this Oncology Models Forum has about, I think, 20 running grants, 4 of them are in breast cancer. And we just received a supplement with them to work with Sean Egan, the chair of this session, to develop new in vitro murine, in vitro models from his murine ILC models. We also have another supplement with Yi Li, who is making rat models of ER-positive breast cancer that we hope to enable as ILC models as well. So copying what Stuart Snit said, I am going to give you the take-home message first. It was posted to bioRxiv last night, so you are welcome to scan that QR code. I pray you don't read it while I'm speaking, but you can take it and you can read it, and everything is on there. We are making all of this data publicly available. I'll show you on the next slide how you can get to most of it. Some of it is not easy to put public, but I'll show you on the next slide how you can get to the mRNA expression data, and then you can get to the whole exome sequencing data, and then we're going to figure out there isn't obvious ways for sharing structural variant data at the moment, but we will make it publicly available. Just to say that if you just take, for instance, SNP array data and compare the genotypes of our cell lines to what other people are using, an important thing to make sure we're all talking about the same thing, they are identical to what was used in one of the largest studies called Makoti. So this is Makoti's cell line, this is our cell line. They are nearly 100%, essentially 100% identical. So we are using, this will be a reference data set that you can kind of reference back to. I should have said, we grabbed every cell line from ATCC unaltered. These are the kind of the true cell lines. So if you want to go and grab the data again, you can grab that QR code over there. It'll take you to our instance of CBio. On CBio, you can then play around with the data. You can access the whole exome sequencing data, the mRNA expression data. This is, Matt said earlier, is one time when you can use your phones, as I see lots of people doing. And so you can then, this is the 17 cell lines. You can look at their mutational status. There's CDH1 mutations in a lot of them, obviously. None of them have estrogen receptor mutations. PIK3CA mutations. You can look at gene expression heat maps. We will then add on the methylation data and as much data as we can load onto CBio, and then you can play around with that. You can drill down into cell lines. So this is the cell line SUM44, which you just heard from Catherine. It then drills down onto, it has 233 mutations, the frame shift in CDH1, a p53 mutation, a frame shift in p53, NF1 splice mutation, et cetera, et cetera. So the first question of these cell lines is, okay, do they express E-cadherin? They should be E-cadherin negative. If they're going to be lobular, that's one of the second definitions is the immunochemistry for E-cad negativity. And so here's some examples here of 3 of these cell lines. You can see they are negative for E-cadherin. The positive control is MCF7, the ductal cell line. And P120 is at the membrane, just like E-cadherin, and you can see it becomes cytoplasmic here. And Patrick gave a great talk about why that occurs and what that cytoplasmic P120 may have unique functions. So these are classic ILC phenotypes, and this is actually one of those ILC-like cell lines that was not classically known to be ILC. So in the paper, you can then show that we do that for all the cell lines. Obviously, they are all E-cadherin negative, as you can see. They all have cytoplasmic P120. They all have the phenotype, the classic phenotype of ILC. The second question might be, okay, what's their intrinsic subtype? Because that's important to research and to therapeutic targets. So because we did multi-omics, we can look at intrinsic subtypes in a kind of a multi-layered fashion. So this is all the cell lines by mRNA, subtypes by reverse phase protein array, so proteomic subtypes, and then DNA methylation subtypes. And importantly, nearly all the cells have a luminal or a luminal HER2 subtype. There's only 3 that are kind of outstanding ones. One is HER2, this is SKBR3. One is basal, the 468, and one is Claudin low, 1187. And this is quite comforting because this is actually the spectrum we see for human ILC, for patient ILC. So this is from this nice paper from Jason just recently. I think, as you all know, and I think someone else showed this earlier today, yesterday, days go quick. Nearly all ILC have a luminal status, very few are HER2, and very few are triple negative. So most of the ILC cell lines have a luminal subtype. You can drill a little bit further down into this, and this is in the paper, but if you look at protein levels, these luminal cell lines express very high levels of estrogen receptor. The luminal HER2 cell lines, which are clustering with both of these subtypes, have decent levels of estrogen receptor. If you do gene expression signatures for ER positivity, you can see very strong in the luminal, kind of strong in the luminal HER2. And this is a indicator of sensitivity to endocrine therapy. They are all sensitive to endocrine therapy, where these 3 here, these triple negatives here, really have no sensitivity to endocrine therapy. So these ICLE cell line models contain many models of luminal disease, which I think is quite encouraging for research. These cell lines genomically resemble human ILC. So this is copy number, DNA from chromosome 1 to 23. Red means copy number is increased, blue means it's decreased. This is primary ILC from TCGA. This is metastatic ILC from Memorial Sloan Kettering's data set. You can see they look pretty similar. This is those luminal cell lines I showed you. You can see if you kind of squint your eyes, they look nearly identical to the copy numbers that you see from the human tumors. The luminal HER2 also looks a little bit different to the luminals, but you can see it looks pretty similar to here. The ones that look different are these three other ones, the odd subtypes, the triple negative, the HER2, and the Claudin low. They really do not show a copy number at all similar to these, and that's because it's really a different type of disease. So these look genomically pretty similar. If you look at the point mutations, they look similar and different. So for instance, CDH1 in red is the primary breast cancer mutation rate, and in a little bit orange is the cell lines. And you can see it's nearly the same. Same for PI3 kinase. This is primary disease over here and metastatic over here. So nearly all of the mutation rates in the cell lines mimic what we see in human tumors, but there are significant outliers. ERB2 is enriched for mutations in the cell lines, something around, probably around growing on plastic that's caused that. And P53 mutations. P53 mutations aren't that common in lobular breast cancer. It's about 10 to 20% between primary and metastatic. Yet these cell lines, nearly every single one has a P53 mutation, which is probably required for it to grow in cell culture. So as long as you know that, you can then interpret your results. The CDH1 mutation spectrum, again, looks pretty similar in the cell lines to what we see in tumors. So this is CDH1 mutations in TCGA all the way through the gene. It's a classic tumor suppressor gene that has a lot of frameshift mutations and nonsense mutations. The cell lines have a very similar spread all the way through the gene, these nonsense and frameshift mutations. These mutations are classic tumor suppressor gene. They have loss of heterozygosity. They have a point mutation on the other allele. That's the classic two-hit hypothesis. You only see that in lobular. You rarely or never see that in ductal tumors here. This is from TCGA. The cell lines, which are on this side of the panel, have that similar phenotype. They have these classic two-hit mutations. One of the interesting things we found in these cell lines is that there are a number that have dual genomic loss of CDH1. They have dual deletion of the gene. Both of the copies of the gene have been deleted. And I'll show you that on this slide here. So here are 3 cell lines that have large-scale genomic rearrangements where this part of the gene has got cut out. CDH1 is completely deleted. This one, it's being cut out here and it's got deleted halfway through the gene. And this one, again, has been completely deleted. So they have 2 copies just totally lost and they don't contain any one. We also found some with a small exonic deletion. So the classic cell line MM134, the most used lobular breast cancer cell line, is unique in its mutation in that it actually has a deletion of an exon, which causes a frame shift. This is relatively uncommon, but we find a bunch of others that have these exonic deletions. And actually, alpha-catenin, which was mentioned by Patrick as well, also shows a dual exonic deletion in the 468 cell line. So we reached out to Ethan Sokol, who's at Foundation Medicine. He's a long-term collaborator and long-term friend who we've published a lot with, and asked, this odd pattern of structural rearrangements, these dual copy loss, how often do you see that in their data set? Because they have one of the largest data sets of sequenced breast cancer. And Ethan is speaking at this meeting later than this. So Ethan found a number of structural rearrangements. We reported this at San Antonio in a poster at the end of last year. Found a number of structural rearrangements in the prevalence of these structural rearrangements, these either exonic or gene deletions. It's about 0.2% in IDC. It's about 0.6% in ILC. It's similar between local and metastatic disease. And importantly, the structural rearrangements are independent of these point mutations. They call them short variants. These are the point mutations I was mentioning earlier, like their frame shifts. Likely, the fact that they are independent of this likely suggests that they're both drivers in their own unique way. So I do think probably one of the underappreciated ways to get rid of CDH1 in ILC is probably these structural rearrangements, which are harder to detect. So we detected those structural rearrangements with a novel platform. I won't go into this. It uses, it doesn't actually sequence DNA. It actually visualizes DNA called optical mapping. You basically take linear strands of DNA, string it out, and then just look at it, and then you can visualize where the DNA's got disrupted. We did this in collaboration with a company, BioNano, who developed this technology. And I should say this was funded by Dyname and Flora. So we've done this on all of them. This is in the paper. So basically, you can look at your cell line of interest, and you can see here, it's got these huge rearrangements where DNA is getting cross-linked between this chromosome over here and this chromosome over here. They are all highly rearranged. Osama then quantified this, so you can quantify, you can pick your cell line of interest, numbers of structural variants. This reflects their genomic instability. SKBR3, the HER2 cell line, being one of the highest. What happens is that these structural rearrangements cause massive damage to massive numbers of genes, causes a whole bunch of new gene expression. It's much more complex than, say, a single point mutation in a gene. You're having this swath of deregulation of your genome. At the methylation level, methylation tends to be understudied, and there's not a lot out on methylation in ILC. These cell lines at the methylation level split based upon their intrinsic subtype. So the luminals are over here, and the luminal HER2s are over here. Osama looked at differential methylation patterns in cell lines and tumors, and found this gene, AP2 beta, which Christian has been studied and has published a couple of papers on, showing that this is hypomethylated and overexpressed in ILC. And we find the same thing in both tumors and in the cell lines. So this is looking at mRNA expression of AP2 beta versus methylation of its exon 1 and its promoter. So when it's hypomethylated, it becomes overexpressed, and those red dots are ILC. So ILC is hypomethylated, overexpressed. And you can see down here, you have the IDCs in blue, which are hypomethylated, underexpressed. And the cell lines essentially look really nearly identical, suggesting that the cell lines might be a nice model for studying and mimicking this epigenetic patterns in ILC. Finally, the ultimate goal of all of this is to look for novel drugs, novel targets, and look at druggable dependencies. And because we did this in a way that we can integrate it with the Cancer Cell Line Encyclopedia and DepMap project, we can then integrate that. So we look at dependencies in number of cell lines, ILC and ILC-like cell lines versus the NST cell lines. You can find a number of dependencies using this that are unique to ILC, and you can drill down and then find pathways. And many of the pathways that we find are ones that we already know about, which is a nice positive control. And Patrick talked about this earlier, about the PI3 kinase pathway, which his group and our group have published on. Seems to be a unique dependency in ILC, focal adhesion, obviously, and then the actin cytoskeleton, which he talked about as well. So finally, we end up with this table of here's the cell lines, here's their molecular subtypes, this is their genomic alterations, this is how similar they represent between the cell line and the tumor at a quantitative level, and then what we believe is their suitability for modeling ER-positive luminal ILC disease. Obviously, that's just our opinion. You can take this data, you can rerun it yourself, and you can interpret yourself, and you can look at your own models and decide which ones you want to use. So to summarize, ICLA is a large multi-omic characterization of breast cancer cell lines with features of ILC. The data is available currently on CBio. You can go there right now and download it and play around with it. And we will make those other platforms available. The ICE cell lines are mostly of luminal or luminal HER2 subtypes and have predicted sensitivity to anti-endocrine therapy, several of them. The ICLE cell lines resemble tumors genomically. The CDH1 exhibits both point mutations, mainly frame shifts, that's the most common, but also DNA structural rearrangements, and we see some of those in human tumors as well. DNA methylation can be studied in both human tumors and the cell lines, and the ICLE exhibit unique druggable dependencies, which can be tested in preclinical models and hopefully will lead us to new drugs and better outcomes for patients. This is the lab. As you know, Steffi and I run the lab together. And I just highlighted Osama because he was the one that really led nearly all of the bioinformatics. But just to say, it was a village that did this. There was lots of people in the lab that grew these cells, that isolated them, that ran them through the core facilities. And I'd like to thank everyone. And obviously, thanks to the people who funded this and also the patients who, if they hadn't have donated their time and tissue, we couldn't do this. Thank you. Yeah, shout's good. I'll repeat it. Yeah, that's a great question. So you mean like a gem? Yeah. Yeah. Yes, totally. So what Patrick's question is, I think one of those fundamental ones is that we have such great tools now to characterize them. How similar are these models to the human disease? And we have been looking at that, for example, in the CDH1 P10 double knockout. And we've been looking at that, trying to do that. It's not trivial to do that. We have a paper just submitted on a novel way to cross-compare, to do exactly what you've said. I'm not answering your question, yeah? I'm just going round and round in circles, yeah. I will, yes, that's our main goal, is to get those quantitative measures out there of what's the most suitable model, and then people can choose from them. Yeah, so clearly there are genomic changes that have developed and they're much greater. Those copy number changes are greater in cell lines. And this has been reported by others. That's not novel. We have done optical mapping now in a bunch of ILC organoids. I think we've done quite a lot of that. And some tumors as well. And so I think we'll get to that point where we can discuss the structural rearrangements and how more there are in cell lines. There will be more. Yeah, so the question was, how many models do we need? Because the disease is quite complex, yeah, and I think Stuart Stint talked about that. The heterogeneity of the disease is a lot, and we obviously need more than a model, yeah. I mean, MCF7s is a dilemma for the whole of breast cancer to be reliant on that for ductal, yeah. I think it's true for lobular. I think some of these, there are like no publications on some of those cell lines, or 1 or 2 publications. I think if people realize that some of those might be better for their subtype of lobular, then more research on them might show that they have better phenotypes. We only use MM134 and SUM44 because they're there. There's no logic for that, out of the fact that they're there.
Yeah, open door question as well. So what do you consider is the best? What should we go for?
We were tried to not say that because then I get shot by the people who are in the audience. I think I would say we will, I think, make some our personal ones of what we think is best on there and in the paper. But I think it's up to the user to kind of make their own guess.
You must have an opinion.
Yeah. And I do hope we will have the paper we've submitted is a platform where you can upload your data and then it will do similarity against human tumors. So you'll be able to do this yourself. That's what we hope to be the final product. Yeah.
Okay, it's my pleasure now to introduce Dr. Demir Barashvili. He's a PI and lecturer at the School of Pharmacy and Biomolecular Sciences at RCSI, the University of Medicine and Health Sciences in Dublin. And we look forward to hearing your talk.
Thanks, John. And this has been such an experience to just see this community in action, in person. So I've really appreciated the invitation to come here and present some of our work as lobular newbies. And, I'd like to thank the organizing committee for putting me after Catherine and Adrian as well. So it's great. But what I'll be talking about a little bit today is our efforts to build on our lobular and in general, our breast cancer research. And what I'll be talking about really is our search for some novel actionable targets. And when I'm talking about novel actionable targets, I mean something else than when people are talking about clinically actionable targets. And when people are talking about clinically actionable targets, they're really referring to mutations that can be clinically targeted. And when you look at clinically actionable targets, the way we look at it as cancer researchers, they vary. They think that you can detect them in up to 30 to 40% of cancer types. And this obviously varies depending on the cancer. It varies depending on the type of tumor that a person might have. The other thing that's really important is that certain cancers are obviously more clinically actionable because these clinically actionable alterations have readily available drugs. So if you look at things like lung and melanoma, where you have many inhibitor against EGF4 and BRAF. The other important thing to note is if something is actionable, doesn't mean that it's effective. It's not going to work for everybody. And this is down to the fact that there's resistance mechanism to this, whether they're both intrinsic or acquired. And what's really important is that we kind of go and look beyond the DNA mutations and go beyond looking at the mutated landscape to find new targets for the patients. And this is really where kind of a lot of our work is centered around. This is around epigenetics, and this is looking at chromatin biology, looking at DNA and RNA methylation. So regardless of whether you're ductal or lobular, the breast cancer cells have a remarkable plasticity, and this plasticity continually evolves from the initial onset of cancer, on treatment, and throughout metastatic progression. And this is again down to therapeutic pressure. It can also be very much modulated by the microenvironment of where this cancer is actually spread or where it's gone. And when we look at alterations, kind of going back to mutations, this is the MSK impact cohort, that they have about 120 ILCs and about 560 IDCs, and these are all metastatic tumors. So these are essentially tumors where the treatments have failed, and they've gone to various metastatic sites. And there's many alterations, and the thing that you can see, and if you look at it specifically in terms of ILCs, looking at frequent alterations, this is basically where you have it present in more than 5% of the ILC populations, what we use as a cutoff. Of those, about 98 are considered cancer genes. And then there's about 43 genes here, which have various different levels, 1, 2, and 3 of actionability. And this, of course, includes EOR, EOR-BB2, your HER2, and the PI3K mTOR. And what's really interesting is a lot of these mutations aren't really, they may be enriched in ILC, but they're not necessarily unique to ILC, obviously with the exception of CDH1, and that's very nicely sort of easy to visualize here. There's a couple of other epigenetically important factors that I would say, and also PI3KC. And obviously, Adrian has mentioned now that the TP53 is really not a lobular thing, but it's very common in IDC. So really, the DNA level alterations don't necessarily fully explain what we see in terms of treatment resistance, what we see in terms of biology, and also metastatic proficiency. So this is really what interests us, to look beyond the DNA mutations. And it's really this sort of data, and also us being very good friends with Steffi, that sort of led us to go and explore in terms of what's happening in the lobular space. And this is kind of a shout out to Siobhan, because I think she'd have some words with me if I wasn't giving an Irish flavor to the lobular. So this is really what it looks like for us. Now, keep in mind, Ireland's about 5 million people, so the numbers may look small, but it's still very relative. So just like in the States, lobular throughout the years have been recorded, and they represent about 15% of all of our breast cancer cases. And really, even though it's 15%, if you kind of break down the ductal and the lobular, because we do believe they're different diseases, if you break them down into different diseases, they are the 6th most common invasive cancer in women in Ireland. So it's only lung, colorectal, uterine lymphoma that goes ahead of it, and obviously ductal. So it's an important issue, and it's understudied in our space, just like everywhere else. And it does pose unique challenges in terms of diagnosis and the treatment itself. So we looked at this, and we decided we kind of wanted to try and see what we can do in terms of our own collections and our own research. So there's a study that's been set up, and that's run by Leonie, who you've met at this meeting, and Arnie, and they've essentially set this observational translational trial, which is called the Breast Cancer Proteomics and Molecular Heterogeneity. And this trial has been running for a number of years. And we have about 1,500 or more patients that have contributed to this study. And what we do with this is we collect clinical data. Patients kindly donate they donate tissue, they donate bloods. And from this, we essentially can create we can create our tissue microarrays for different primary and metastatic tumors. We try to do a lot of cancer modeling, genomic profiling. We have liquid biobanks, and then also we do a lot of translational studies where we try to make novel models. And we've created this tissue microarray from this study. And in this tissue microarray, about 900 breast cancer primaries, of which 124 are lobular, which is what we use for a lot of our current studies. And I will mention that a lot of the stuff that I'll present to you as well is kind of early stage studies, and we're sort of still trying to kind of get to the bottom of what's happening. And another thing I'll mention is when you look at these, when you look at the actual patients that have contributed to this study and to our trial, you can see that just if you look at overall survival, that the lobular patients actually end up doing worse over time. So we have a very long follow-up at this stage. It goes up to, most patients are covered up to 20 years. So even though this isn't significant, you can see that generally speaking, that the overall survival and overall survival is actually worse in lobular, which is kind of in line with what's been reported. So what I'll go into next is kind of moving away a little bit from how Adrian has presented, looking at many different models. What we're trying to do, we were trying to create patient-derived models from the donated tissue. And what we're trying to see is looking at essentially this transformative power where we can apply N-of-1 studies. And they're really good in the sense that it gives you a powerful microscope into individual cases, because we keep talking about heterogeneity and how each patient's tumor is different, really. So we're trying to see on an individual level, what can we figure out in terms of precision medicine for an individual tumor. And every patient that we can actually create models for is essentially represented by their own avatar, for lack of a better word, and gives us unique research opportunities. And this is our workhorse model for lobular. So this is a model that we created. Initially, it was on our study as a primary lobular, hormone receptor positive, gone through the usual treatment, chemo, radiation, tamoxifen, Zamida. There was a metastatic recurrence, and it was the first recurrence, and it was to the brain. And from this model, it was biopsied, and we were able to make the actual models. And you can see here, this is estrogen receptor in the primary tumor, and then this is the metastatic tumor that was biopsied as well. And you can see there's no ecadering. And from this, we created PDXs. We created organoids from these PDXs, which we use as our model. And this particular model has resistance to SERMs, SERDs, and also CDK4/6 inhibitors. And when we looked at the DNA profile, there's no real easy actionable mutations, for lack of a better word. So there's no EOR, there's no EOBB2 mutations. And it makes it a really good model for us to kind of go and look into whether there's anything else that we can go. So looking again beyond the classical mutation profiling. And this is just this model in vivo. You can see nicely that it still remains estrogen responsive. So using this model, we've managed to get a lot of different hits, I would say. I won't be able to talk about all of them, but I've highlighted some of the different epigenetic vulnerabilities that we found in this one particular model and in this one patient-derived model. We've been able to see that there's a chromatin remodeler and transcriptional coactivate AIB, which is very important in ER-positive tumors. We see that it's sensitive. You can inhibit AIB1, and we can see as well that high AIB1, this is a metabric based on gene expression cohort. High AIB1 predicts worse survival in patients. We've seen a number of other things, including DNA methylation. DNMT inhibitors are effective in this particular model. CDK12. It's a kinase. We've noticed that this is, again, we've stained our tissue microarray for CDK12 protein expression. And while these aren't significant, you can clearly see that high CDK12 expression lobular patients do worse in terms of overall survival. And if we use a CDK12 inhibitor, we can get a nice response. And I should mention that these are actually patient-derived organoids that we made from this model. Similarly, RET is another thing that we work together with Steffi and Adrian on. And this is, again, in the absence of these fusion or in the absence of genomic alterations, we see that in lobular models, if you stay in RET, you can see that the lobular patients with high RET do worse. And if we treat it with a LOXO inhibitor, which is now available and it's FDA approved for certain tumors, you can get a response in these models. But what I really want to talk about briefly today is kind of looking at two different aspects that we found that are kind of a little bit different. One is based on DNA damage, and I saw from Matt Sikora's lab, they had a nice poster as well about PARP inhibition in lobular. So I think that kind of synergizes nicely. And then finally, I'll talk a little bit about another layer of epigenetics in terms of RNA methylation. So these models don't have BRCA1 either somatic or germline mutations. But what we found when we did a study with Steffi and Adrian and Fergus Couch is that we could detect these HRD mutational signatures, which is also known as signature K. And this HRD mutational signature essentially is if you're negative for this particular mutational signature, you're considered HR competent. So if you gave it a PARP inhibitor, you still have active homologous repair, active DNA repair, and the cell is able to survive. We managed to detect this HRD mutational signature, which makes the cells deficient in DNA repair, meaning that if we give it a PARP inhibitor, we can actually get synthetic lethality and we can get kills. And we detected this particular HRD signature in our ILC model. We also detected in IDC models, but it seems to be specific more in a very small cohort of ER-positive metastatic tumors. And again, if we take this model, knowing that it has this HRD signature, if we treat the organoids with niraparib, we can see a really nice response to the drug itself and the organoids. And also, if we take these cell lines and do an in vivo study where we give it niraparib, you can see again very nice response with the niraparib treatment, both in terms of the primary tumor, and I'm not showing it here, but we also see a reduction in the metastatic tumor. So this is another avenue that we can kind of explore for lobular patients. So these are the ones that do not have BRCA1 mutations, germline or somatic. But if you can go and find these signatures where you can see DNA repair deficiencies, it opens up possibilities for further targets. And finally, I'd like to touch on another complication in the epigenetics field. We heard yesterday from Renata a really nice talk in terms of chromatin biology. We've done some work on DNA methylation, which is what people mainly talk about. But now there's another thing that's really emerging, and this is RNA methylation. And essentially, we've done some studies, the studies that have come from Leone's group, and basically we found that there was enhanced role for RNA methylation, in particular in terms of endocrine resistance and in terms of metastatic progression. There was these dynamic shifts that we saw in the RNA metallome as the tumor progressed from the primary to treatment resistant to metastatic. And we used lots of different sequencing methods, including M6A RIP, which is basically kind of similar to the ChIP-seq stuff you heard about yesterday. We used transcriptomics, proteomics, all sorts of multi-omics to essentially map out the RNA modifications across the different progressions. So if you're interested in that, I'd suggest you look up this paper and you can kind of see you can get a better feel for how we came about to identify M6A as important in the context of endocrine resistance. And one of the things that we found was FTO, which is actually a key regulator of RNA methylation, and we found it as a potential target in these poor responders. And we initially identified it in IDC, but we decided to go and also look at it in terms of our lobular patients. And again, when we stain FTO in our tissue microarray, We see very nicely, this is progression-free survival. We see that high expression of FTO indicates much worse outcome for lobular specific patients. And again, FTO has nice available inhibitors. So we did a study using the MIND model, which Catherine's described very nicely earlier. So I don't have to go into it. I don't have to go into it too much. But we essentially took the T638 cells, or lobular cells, we injected them intraductally, and then gave them the CS2, which is an FTO inhibitor. And again, we see a really nice response in terms of primary tumor, and more importantly, we see a really nice response in terms of metastatic disease. And when we profile these treated tumors, we actually see that inhibiting FTO, it kind of more modulates the actions of FTO rather than seeing some of the ECM components that we've heard about today. But we see reductions in MYC, cell proliferation, and sort of metabolic pathways. But the drug works really well, and it actually has a very good efficacy in the lobular models themselves. So finally, what I hope I've communicated to you is that there are lots of interesting things that are actionable that go beyond DNA mutations, particularly in terms of epigenetics, particularly in terms of RNA methylation, which is a big interest to the group. And these N-of-1 studies, they serve as a very powerful microscope in terms of allowing us to zoom in into individual cases, and we can uncover these unique vulnerabilities that might necessarily be shared across everybody, but we can kind of start building out individual stories, which is kind of the true aim of personalized medicine. So we've identified these vulnerabilities in kinases like RET, CDK12, the HRD pathway seems to play a role. These epigenetics drivers like AIB1 and FTO are another new therapeutic avenues. So what we need to do next is obviously deepen our understanding of these, and we want to basically try and expand and get involved with more lobular models and try to validate some of our findings. So this is a thanks to the larger team. Obviously, a lot of the collections and a lot of the work that I presented here is sort of a joint effort with my partner in crime, Leonie. So I can't take all the credit. Many collaborators, people from the labs, our funders, and of course all of our patients, and of course we have our very own Lobular Ireland as well. So we're well represented by Siobhan. Thank you very much, and I'm happy to take any questions.
Damir, that was beautiful. I was curious, in your experiments with the AIB1 inhibitor, given the really tight crosstalk between AIB1 and the estrogen receptor, and then thinking about the data you showed just before that, where they're really resistant to all of these agents, but there's a pretty profound estrogen response. Have you looked at whether that AIB1 inhibitor affects estrogen responsiveness in conjunction with its inhibition of proliferation? So we haven't looked at it. We kind of looked at it more in the context of the endocrine resistant pathways. So not necessarily, it doesn't have that clean sort of estrogen response signal that we would see in terms of some of the early growth factors, really clean GREB1 and PS2, but it seems to be working through a sort of a hybrid, where you're getting that hybrid model between estrogen signaling and the growth factor pathway. So even though AIB1 is there, and obviously if you treat even endocrine sensitive cells with AIB1 inhibitors, you're gonna kill the cells. But it seems to be, depending on the context, it has a slightly different role, and we've identified different partners. So when we pull out AIB1 and we kind of have a look to see what it's interacting with, we don't actually see EOR there in this model. So there's no EOR, there's no FOXA1, there's none of that the core complex that you would say kind of has completely new partners that kind of throw it more towards the pro-metastatic phenotype.
This is very inspiring work that you're doing. As a patient advocate, my question is, do you foresee, because each patient, as you said, is his own research project, of applying this to individual patients, because how long would it take to be able to investigate all these different avenues on one patient?
Yeah, and I suppose one of the caveats is, and it's been highlighted as well in Catherine's talk, is that we can't make successful models from every patient, especially when it comes to the estrogen receptor positive disease. In terms of, let's say, making the patient-derived xenografts, there's still about 30% take rate. Organoids are a little bit better, but it gives you, it's such a small, it's still a very small percentage. So we're trying to build, the idea is to build it out on the people where we can make it, and then try and find some ways where we can extrapolate it to the larger population. And these are very early studies. And it's about really finding out, where does this apply to and who is it relevant to. Well, for one particular specimen, so let's say in vivo, to set up the model, so in vivo being the mouse, it will take anywhere between 4 to 5 months from when you get the sample. And they work best in terms of metastatic samples. So sometimes, 4 or 5 months is a very long time in terms of metastatic diagnosis. Organoids are a little bit faster. If you go straight from the tissue to the organoid, you can get a much better, you can get a much faster output in terms of, they probably take about 2 months or less even to set up and actually do a lot of the testing for the sequencing and testing. So I think it's a very emerging field. It's still very early, but I think there's lots of hope, and a lot of good people are working on it that are much better than myself, that are trying to make this thing work. Thanks, John. Thank you.
Our last talk in this session is by Christina Gersh from the Department of Internal Medicine, Division of Hematology and Oncology, University of Michigan Medical School. And she was selected to give a presentation on the basis of the poster number 13, which I happened to be judging yesterday. I was very impressed by. So look forward to hearing your talk.
Yeah. And let's choose the caller. And then you can put the acting.
Can you hear me okay? Yes? Okay. So first, I just want to say thank you for the opportunity. Oops, I'm already messing up over here. Thank you for the opportunity to be here today to discuss our lab's research abstract. I'm going to be talking about the lobular cell lines we've just discussed, the two that are the most popular to use, mortalized breast cancer cell lines, and the resistance mechanisms to CDK4/6 inhibition and endocrine therapy. So as people, this is very sensitive. As people have said, there's a limited number of cell line models to use in lobular. So in blue here, we have the MDAMB134s and the SUM44PEs that we used for our resistance models to generate models to CDK4/6 inhibitors and endocrine therapies, the treatments you see here in the yellow table. We also generated a lot of ductal models to compare. We're still receiving our RNA-seq data now. They're all estrogen receptor positive immortalized breast cancer cell lines with varying degrees of ER/PR HER2 status. So our lab has been studying resistance mechanisms to hormone therapies and CDK4/6 inhibitors for the last couple of years in lobular breast cancer most recently. We're using hormone-free conditions to mimic aromatase inhibitor treatments, along with tamoxifen and fulvestrant. We're using palbociclib, abemaciclib, and ribociclib in our experiments, but today I'm going to focus on palbo. Our main goal is to uncover mechanisms of resistance to CDK4/6 in endocrine therapy in order to identify strategies to overcome it, and also have cell line models to use potentially for our group and other groups in the future. The main mechanisms of resistance to endocrine therapy involve, these are just generally the loss of estrogen receptor or varying ESR1 mutations that cause estrogen-independent cell growth. Efforts to investigate mechanisms of resistance to CDK4/6 inhibitors are ongoing. Here I listed some of the main types being reported and also that we've seen in our own cells that I'm going to discuss here today. Oops. Okay, so first, this diagram is a depiction of how we established our lab's models of resistance to palbo. We used two different methods. On the left, we dose escalated the amount of inhibitor from 10 nanomolar over a long period of time, 3 to 6 months, until we reached a peak dose of 500 nanomolar palbo. We call these cells the acquired resistance or AR cells. This took a very long period of time in the lobular models because they're so small and they grow so slow. On the right, we used a different method and we hit the cells with the highest dose all at once, 500 nanomolar palbo. We call these the intrinsic or IR cells. Our hypothesis or reasoning for using these two methods was to try and discern whether varying resistance mechanisms might occur based on what was already inherent in the cell, intrinsic, or what might develop over time, acquired. And then on the right here, we have a dose response graph with our lobular cell line, the M134s, and palbo treatment. The black line there represents the parental cells with an IC50 value of approximately 45 nanomolar. The IR cells in red and the AR cells in blue are shifted further to the right because they've become less sensitive to palbo with all of the splits and maintenance. So then this slide is showing you our endocrine resistant models and how we establish those. So we took confluent flasks of parental cells, wild type, and we put them in 3 different conditions seen here. On the left, we have hormone-free, which is meant to mimic an aromatase inhibitor condition with limited estrogen in the media. We also have the fulvestrant and tamoxifen conditions, which were also maintained in hormone-free media. On the right, we have a dose-response graph showing you MCF7 cells that were treated with estradiol. So in black, you can see the parental cells are highly sensitive to estrogen with a 300% change from control at the max. And the cells that have been maintained in our 3 endocrine therapy conditions are seen in the blue, red, and green. They have a limited growth response to estrogen, as we would expect, because they've been maintained in hormone-free conditions and are largely estrogen independent. Oops. Okay, after we took the time to generate all of these models, which took quite a while, we confirmed a change in growth pattern by dose response. I didn't show you those here. We collected RNA from each cell line and sent them for RNA-seq analysis. Just pointing out that we also sent the parentals for comparison, as well as a short-term 3-day treatment to help us decipher differences between short-term and long-term resistance. All right, so for the purposes of the short talk, I'm just going to discuss some of our preliminary RNA-seq data that we received back in the SUM44s as an example. So this graph is showing an upset plot from a meta-analysis using the molecular signature database, the hallmark signature pathways. In pink, we have the cells that have been treated with 500 nanomolar for a short duration of time. Some of the most significant hallmark pathways are listed in this table with the p-values to the right. The top 3 pathways were all involved in cellular proliferation. And considering these are CDK4/6 inhibitors that cause disruption in cell cycling, we would have expected something like this because they're the most affected pathways in response to a short-term treatment. Very sorry. All right, so let's talk about the IR cells in yellow. So the top hallmark pathways are listed again with p-values. Estrogen response late and early. EMT, hedgehog, and KRAS signaling suggest different resistance pathways are occurring in the long-term adapted cells versus the short-term that I just showed you. And then in blue, we have our AR cells with the top signature pathways. Here, interestingly, the most significant pathways are also involved in proliferation with E2F, G2M, and mitotic spindle. This is possibly because they're in the earlier stages of resistance, similar to the short-term treatment. They're still kind of developing. But interestingly, want to point out that EMT and estrogen response late are also top signature pathways, suggesting that these actually may be long-term resistance mechanisms involved in this lobular cell line in response to CDK4/6. So then we kind of tried to dive into some of these pathways to start and look at some of the common EMT gene markers. So N-cadherin on the left, vimentin in the middle, and fibronectin on the right. These are expression levels from our RNA-seq with blue and yellow bars representing our long-term AR and IR cells versus the short-term treated in pink, which you can barely see because there's very little change. This hints at potential EMT mechanisms of resistance to palbo in this lobular cell line, and we're in the process of confirming protein levels by Western and designing other experiments. This is just our preliminary data. So next I want to focus on the 134 cells. In either the palbo condition on the left or the endocrine therapy conditions on the right, we saw a great deal of androgen receptor upregulation at the gene level for all of these growth conditions. This response has been reported by other groups, but we've not seen it in lobular cell line models yet. We also checked the protein level expression of AR in the palbo models and confirmed it is upregulated. We actually have 4 models of resistance in this cell line. We have the 300 nanomolar and 500 nanomolar because we had such a hard time getting them to grow in 500 nanomolar. We started with 300, and in the end, we ended up with 4 models. So that's better than 2. But we also checked the protein levels in the endocrine conditions. I don't have the Western shown here, but it is up in those as well. And this is exciting because AR is a targetable protein, and we plan to test these models with various androgens and anti-androgens in combination with CDK4/6 to see if we can resensitize the cells. Okay, so in the same lobular cell line maintained in endocrine therapy conditions, we also saw an upregulation of ERBB2 on our RNA-seq analysis seen on the left. This gene encodes for the HER2 protein, which we all know. We saw an upregulation at the protein level in all of these growth conditions. We're in the process of testing anti-HER2 drugs to see if we can reverse growth in this set of cells. I'm also interested in looking at sequencing these now because I had a talk yesterday where we were talking about HER2 mutations. Potentially those could be there as well. We're currently testing our models against other treatments and checking for cross-resistance between endocrine therapy and CDK4/6 inhibitors. In the example shown here, we took the 134 endocrine therapy cell line models and treated them with palbo. The parental cells are shown here in blue. All the other cell lines appear to be slightly less sensitive to palbo after undergoing endocrine resistance, but we have a lot more work to do, a lot more IC50s, dose responses, growth assays to check. So in conclusion, we have generated multiple breast cancer cell line resistant models, lobular, to use for future experiments. RNA-seq has identified novel pathways of resistance in these lobular models, including potentially EMT, androgen receptor upregulation, and HER2 upregulation. Our future experiments will focus on targeting these pathways and other drugs or combined therapies to attempt to restore sensitivity. I just want to say thank you for the opportunity again to present our lab's research. Listed is our team from Dr. Ray and our collaborators, and we're very grateful for the generous funding from Dynami and Flora and the Rogel Cancer Center. Thank you so much.
The androgen receptor, I've been listening yesterday and today, and I don't see the androgen receptor. I'm a surgeon from Buffalo, Roswell Park, and I'm a big candidate for making sure that androgen receptors are part of the testing because it's an option that's actionable. And I'm glad to see you're presenting your data here today. I was just wondering if you're using any enzalutamide in your lab at all with the androgen receptor?
So before
blockade.
Before I left, can you still hear me? Before I left, I tried biclutamide and DHT, and I was not the greatest dose response, so I'm still trying, but I'm going to write down the drugs that you're recommending here. So we have, I don't.
It's an inhibitor. It arrests growth, so that's good. So patients can stabilize their condition versus a progression. So using the prostate literature, since it's also, and I'm just going to jump off line here, E-cadherin in prostate cell cancer lines also. I think enzalutamide is a great option, especially as we go through the clonality and the changes that we see with recurrence and metastatic disease. I really think all our women should be getting AR testing. And I really appreciate you bringing this here because it's opening our eyes.
Thank you. I appreciate your comment, and I wrote down what you had to say, and I'm going to read more. Thank you.
Okay, I think we will go ahead and start. This, I should say that I am like super nervous. It seems like I'm more nervous than for a scientific talk. It's a kind of a different session, but we thought this is most appropriate. I think a lot of people have commented that this meeting is great. We have a equal distribution, equal attendance from physician, advocates and scientists in the room. It's really, really great. And I should say, and I think most of you know that a lot of this has come together because of Lee Pate, who unfortunately passed last year. But she has shaken up so many things that we thought we'll have a session here where a few of us who were close to her, who were directly working with her, affected by her, with her, would say a few words. I also want to say at the beginning that we are very fortunate to have Lee's mom here with us, Mary Pate. And Mary, I don't know if you want to stand up for a second, but anyway, if you haven't had a chance to meet Mary, please meet Mary. So I'm going to start off just quickly again. The first time we had the symposium here was 2016 here in Pittsburgh. I want to mention Dr. Nancy Davidson, who at the time was the director of the Cancer Center here in Pittsburgh and who gave us tremendous support and encouragement to really start this. On this slide, you see the picture of the patients, the advocates who attended the meeting. There were many, many fantastic patients, advocates there, but really the two who stood out, and some of them are here now actually, the two who stood out really were Lori and Lee. And there was such momentum that after this, I had a few phone calls with Lee who said, okay, we really need to do something. It's changed my life, the cancer diagnosis. I want to do something. What can we do? Multiple phone calls. She took action then, or the lead, I should say, in writing the white paper. A lot of us who were at the meeting there helped. But because of her skills as a prior consultant and writer, she had a tremendous gift to actually for fantastic writing. We developed a logo, and I think Daniel Brown is in the audience here, who worked with Lori to develop the logo for LBCA. And we said, yeah, let's get something off the ground. Daniel also helped them to start the website, and we did an abstract submission for San Antonio 2017. So in 2017, we had then the first meeting really of LBCA. We made a poster. We put the first poster up from LBCA, and you can see this here on the bottom left, yeah? You recognize a few faces, again, Lori and Diane, our great moderator from the social media yesterday. And yeah, I think Lee, one thing there were so many great things, but one thing, she really was tremendously inclusive. She was always asking patients, scientists, physicians for feedback and really incorporated it. Yeah, it was really, really stunning. You can see Rachel Jankowitz was on the advisory board as well. And yeah, there was tremendous movement, action, and everything. So in 2018, just a year later, I think we had gained enough traction. There was a mini symposium at San Antonio where we summarized the current state of lobular breast cancer, the clinic as well in the research. And as many of you know, this year, there's going to be another session at San Antonio with a number of talks from folks here in the audience. So, and actually this will be the first time where we have a patient advocate. Siobhan is actually going to be on the panel. Lee, I already mentioned, Lee was always for involving everyone and for moving forward. But in a focused way. She really tried to say, okay, what are our most important questions? How do we do this? And at one point, she worked with Susie McDonald, who's also in the audience, and we got together in Cleveland. We got together in Cleveland and thought, okay, so how do we move to the next step? Who do we need to include? What do we need to do? And this is actually the poster on the top right. Janice Axelrod is here. I think there are more people here in the audience, but yeah, saying this is what we need to do. And out of this, when we wrote or I should say Lee took charge of writing an article, how researchers, clinicians, and patient advocates can accelerate lobular breast cancer research. Many of you here, Christine is in the audience, Laurie, Claire, Siobhan, worked together on writing this. And I summarized here, and I just want to highlight a few examples. So define ILC research priorities. We did this global ILC survey. The paper is going to be in people's inbox tomorrow, I should say. We finished writing it, so you will see it tomorrow. Design clinical trials, Gelato, Rolo are examples. Standardized diagnosis of ILC. We saw a brilliant talk from Dr. De Shepper yesterday on that study they did, and obviously the keynote from Stuart Schnitt was absolutely outstanding. Collect data and samples. We work together with folks in the Great Lakes Breast Cancer Consortium to do a really large data analysis of 30,000 patients and 3.5 patients with ILC. And also, and you will hear more about this, a BCRF legacy project. Dr. Jagmohan Huda is going to talk more about this. Another point on the top right was the assemble and communicate educational and research information on ILC. I think there's really so much out now, ELBCC, LBC, Lobular Breast Cancer UK, Lobular Ireland, and, social media, so much out there now, right? Integrate or show integration of ILC into cancer conferences. I already mentioned San Antonio, the mini symposium. There was first time ever, and I think Christine was really heavily involved, this a presentation at ESMO, and then again at San Antonio this year. Support, foster worldwide collaborations. I think I showed a picture of GLOBMOB earlier, that we really, GLOBMOB stands for Global Lobular Mob, right? And Diane came up with this. That's exactly right. At first it was LobMob, and then suddenly it was GLOBMOB, right? So, and this is a group of patient advocates really representing the different groups, also independent advocates from all over the world, because we don't want to reinvent the wheel and really work together. And we think that, we can do this. We can get together for these yearly meetings to prepare, but then also to analyze and improve. And I think that would be great. And then finally, encourage research funding focused on ILC. And again, Diane talked about this yesterday. There's lots of things happening over the last few years. This is a survey. It was shown at San Antonio next year. And again, the paper is going to come out soon. This was something which was really, really close to Lee's heart. She really said, okay, let's just try to figure out together what are the bottlenecks? What do we need to do to really improve lives for patients with ILC? And we surveyed patients and physicians, researchers, identified a few topics. And please, the paper is going to be on bioRxiv in a few days. Read it carefully, yeah? Lee did not care for recognition. That's the last thing. She would always sit in the back. That was not something, that's not the reason she did anything, right? She did, but it was recognized. Her efforts were recognized. Examples are here. She received the Best Cultural Advocacy Leadership Award at the Northwest Metastatic Breast Cancer Conference in Seattle. She became a Komen Scholar and many more, but this is just two things I'm going to show here. And again, one of the things she really promoted, which was tremendously important for her, is to increase ILC advocacy, not only nationally, but also internationally. We have a couple of groups now represented here on the front, and I think all of you guys can really attest to how much influence she had in this. And there are others which are either embedded in larger organization or others, not even mentioned here. I mentioned quickly, Susie, we talked about this, within the Great Lakes Breast Cancer Consortium, we are starting to get an ILC advocates together. So, again, a lot of this is really true to her initial efforts. I should say, in addition to all of this tremendous advocacy, she was also a very close friend. She always saw the glass half full. She was always fun and great. I also learned this strong German, and I said I wouldn't tear up here, and I try not to, yeah. So we don't really need much to like think about her and remember her and stuff. But one thing we did actually, she was also a great photographer. She really took absolutely beautiful pictures. Upstairs in our lab, we have a wall where we have some of the photos she shared with us, Mary shared with us, yeah, from her trips through India and other places. The other thing, finally, at her celebration of life, I learned from some of her friends in Seattle that her favorite cocktail was The Last Word. So if anybody wants to know, this is equal parts of gin, maraschino liquor, lime, and green chartreuse. It's really, really good. It's really good. We actually, some of the original LBC advocates, we actually had a, what do you call it? Yeah, a virtual drinking. What's it? We all toasting. Exactly. We all bought the ingredients and we toasted to her, and it's really good. And if I'm not well, I take all these ingredients out and have one, and then I'm better. So this is a picture at the celebration of life in Seattle, and Mary had actually asked me to write something for the obituary, and this is part of what I wrote, and I just want to read this, and this is my last slide. Lee was tremendously generous with everything, her time, her resources, and her energy. Her ability to bring people together with a common goal led to lobular breast cancer being recognized as a unique disease that deserves further research. And it initiated many national and international collaborations among researchers, clinicians, and advocates. These contributions to lobular breast cancer advocacy have affected the lives of thousands of patients and their families, and this number will only continue to grow. We are very lucky to have had her in the breast cancer advocacy world. And Mary, I just want to say again, thank you for sharing your daughter with us. She was like absolutely, absolutely amazing. Thank you.
Well, thank you. I can breeze kind of through the history of the LBCA now because Steffi talked about a lot of the important points, but I do have a slide, maybe. Sorry. There we go. That does show a little of the timeline. I met Lee in September of 2016 at the first International Lobular Breast Cancer Symposium, and I think I had the same impression that everyone else did, that she was a natural leader, she was dynamic, she was focused, she was committed, and she was just frustrated enough to want to make change. And she went back to Seattle and she wrote the white paper, but she didn't do it in a vacuum. She reached out to all of us and said, this is what I want to do. What do you think? Give me comments, edit. Will you sign your name when I'm done? And then she sent it to Steffi and she sent it to Dr. Jankowitz and said, is it scientifically accurate? Is it medically relevant? Because I want it to be important. And it was. By January, we were holding regular meetings. Lee was our leader, and I was proud to be her deputy. In February, the BCRF sent out to their vast newsletter list an article about the symposium, and they talked about the advocates, and they talked about the white paper, and it was such a huge endorsement to the lobular community. By April, we had a logo, we had a name. In August, Lee and Steffi had identified and recruited a terrific scientific advisory board. And in December, we went to San Antonio with our poster. And before we went, Lee looked at all the organizations that were going to be there and sent them an email and introduced herself and said, we're going to be in San Antonio. We have a website. We have a poster. Can we have 10 minutes of your time? And when we got there, everybody met with us and we talked them about what we wanted to do and who we were. And people walked by the poster and we grabbed them and talked to them about lobular. And at the end, Margaret, Margaret came from BCRF and did a live Facebook feed that was really well received and it just gave us a lot of buzz. I think at the end of the conference, we were a little giddy. I remember sitting in Lee's hotel room. We popped a bag of microwave popcorn in her microwave, and we opened a bottle of wine, and we kind of looked at each other, and we said, we have a lot of work to do. And we did. The next 2 years were really the fundamental laying down the roots for the Lobular Breast Cancer Alliance. In February, Diane Mapes, double whammy, wrote the article for the Hutch. It was very well received. A lot of people read it. In August, our research library was established, still going very strong. And then Lee reached out to all those organizations that talked to us and said, what about a webinar on ILC? I can get you a clinician. I can get you a researcher. I can get you an advocate. And they said, okay. And we did a lot of webinars. This was the days before Zoom. And after each webinar, we heard the same thing. We've never had so many people sign up for a webinar. We've never had so many questions after a webinar. And we never knew what we didn't know about lobular breast cancer. She reached out to conferences and said, let's put ILC on the agenda. I can get you a researcher. I can get you a clinician. I can get you patient advocates. And we did. We got into conferences and we talked about lobular. I think Steffi mentioned the Komen Northwest Metastatic Breast Cancer Conference was very near and dear to her heart. And she was, they always had a robust lobular discussion. We went back to San Antonio in 2019 with another poster. And when 2020 started, we were really jazzed to help to start organize, which would have been the fall 2020 Lobular Symposium, again here in Pittsburgh. But as everybody knows, COVID came and life changed. And coinciding with that, Lee's health took a priority in her own life, and she stepped down from the Lobular Breast Cancer Organization. She wrote us all a really beautiful email, and it was, I'm so proud of us. Look what we did. We went here, we did that. It was never, look what I did. That was her great strength. She was so collaborative, and she knew that it took a team to do anything. But even though she stepped down, she did not step back from advocacy. She mentored a lot of the advocates here on this panel. She was a prolific and gifted writer and speaker, and she continued to write and speak. She served on the steering committee for Komen Advocates in Science. She was very prolific on social media. I know many of you who've never met her on social media. She engaged with researchers and clinicians and patients, and she continued to work on research grants and push researchers and clinicians for their research. In October of 2021, she wrote a beautiful blog in honor of some lobular breast cancer patients who had passed away. Joyce Bittinger, Meg Kotler, Elaine Eigelman, and Elizabeth Vigiano. They were all here with us in 2016. I did it too. At the end of that blog that Lee wrote, she left words for all of us. She said, and when I die, I am comforted, trusting that other advocates will do the same for me. They will take the seeds of change and progress I have helped sow and help them germinate and thrive. And that's why we're here. Thank you.
I think that's a yes. You can hear me okay? Yeah. Excellent. Thank you very much. It's very moving. Talks about our dear friend. I'm Diane Mapes, double whammied. I actually taught Lee how to tweet. It was not easy, but Lee was an extraordinary person. She was a political consultant. She got Bill Clinton elected, yet she did not play politics. She made dramatic changes in the world of cancer advocacy and research, but she was not a drama queen. She loathed drama. She had little time, 55 short years, yet she was the most calm, patient person you could ever meet. I met Lee through writing. She'd done, as you've seen, a lot of traveling and cycling. Yeah, there we go. It's okay. Yeah, traveling and cycling and all over Ireland and India and Italy and beyond. And she wanted to publish her travel essays and her photography, so she got in touch with me. I was a freelance writer in a former life, And she wanted to talk markets with me. So we talked markets, and we also talked lobular breast cancer, because since we'd both been diagnosed, we'd been trying to get the word out as much as we could about a breast cancer nobody had heard of, that didn't make lumps and didn't necessarily show up on mammograms. That's an issue. So I'd done a bit of advocacy on Twitter and through my writing, but Lee was a power player. She was a born advocate. And she wasn't just perturbed by Lobular's low profile at the beginning. She also took issue with lymphedema and how it was covered or not. And she went to Washington, D.C., her old stomping ground, and she lobbied with Elaine Egeman, another ILC patient advocate, to push members of Congress to pass the Lymphedema Treatment Act, which passed in December, Lee. And the treatment that goes into effect in January. So I work with Lee a little bit on that effort, and I work with her much more on the Northwest Metastatic Breast Cancer Conference, which launched in 2016 with the help of Komen Puget Sound and a lot of METS patients and allies. And lobular advocate Joyce Bidinger, or Joyce Nadine, and Lee carved out a space for ILC at the conference in its second year. And it was a huge deal for all of us. Our breast cancer was finally on a main stage somewhere. That was where I met Steffi in 2017. And we all immediately dubbed her our Lob Mother. In 2018, as Steffi mentioned, Lee was awarded the Beth Caldwell Breakthrough Leadership Award for all that she did to put lobular on the map, including the launch of LBCA. From the moment Lee was diagnosed, she leaned in, she rolled up her sleeves, lymphedema and otherwise, and started identifying pain points in the system where research and care were falling short. She was a visionary with the calm, calculating patience and diplomacy needed to move mountains, or something equally difficult, get researchers to listen to patients. Lee not only got researchers to listen, she inspired them to switch gears, to lean in. As Steffi said at the memorial, Lee Page changed my life. And she also changed research. In just a few years, Lobular's gone from an ASCO backroom slide presentation on uncommon and rare breast cancers to multiple international ILC symposiums, a new European lobular consortium, and soon a major stage presentation at the San Antonio Breast Cancer Symposium. And Lee was a big part of that shift. She knew how to negotiate, to cajole, to plant a seed and then sit back and watch it grow. And she had that patience, even as she was running out of time. As advocates, the two of us were a study in contrast. I was and am ever impatient. I'll be at work at Fred Hutch and I'll see a couple metastatic breast cancer researchers come down for lunch and I'm like, get back in your labs and cure cancer. Lee had a much more subtle, diplomatic approach. Her approach worked. She studied the science, she learned the landscape, she got to know the people. And then she started helpfully pointing out areas that needed some improvement. She got people to listen, and she was ever grateful to Steffi and to all of you, so many scientists who were willing to listen, willing to discuss. She knew how difficult it was to walk new ideas down the closed off corridor of settled science. And when she got frustrated, she would just keep pedaling, just as she did in India when she fell off her bike and broke her collarbone on a cycling trip. She got back on and kept going. As Steffi mentioned, she had no ego. None whatsoever. And she was far too humble to ever acknowledge her impact, but others have. Nancy Davidson compared her advocacy contributions to those of NBCC's Fran Visco and Rose Kushner, the journalist whose advocacy efforts finally put an end to those horrible breast surgeries without a consent, where a woman would go in for a biopsy and would come out with not much, radical mastectomy. Hannah Linden also described Lee as smart, practical, persistent, and noble. She used that word half a dozen times. We would like to have 40 to 50 people like Lee, she said. She saw the issues and sought to address them in a positive way. And she was able to push for changes that were realistic and doable. I was lucky to know her. I really miss her. We were all lucky to know Lee, and we all really miss her. But she left us a legacy and a list. I call it the Lee list. It's a document with all the different ILC organizations and the funds and the consortiums. Lee wanted people with this disease to know where they could get good, solid information and where they could send money to fund lobular specific research. She wanted a lot of things. She cared about a lot of things. And if you go back to her writing, to her blog posts, you could see what truly mattered. As Lori mentioned, her friends mattered. And honoring her friends that had passed mattered. She wrote that lovely tribute to Elaine and Joyce and her dear friend Elizabeth Vigiano. Collaborating and helping others mattered. She posted a lengthy list of community and patient advocacy groups at cancer centers across the US to encourage and nourish patient-engaged research. Travel mattered to her. In one of her last posts, she wrote about her frustrations of dealing with cancer during a pandemic. I wanted to live life as much as possible in the time I had left, traveling and exploring, spending time with friends, walking every day, being in nature, and most importantly, doing my best to contribute something constructive to the world to try and make things better for others. All of you mattered to Lee. Her very last post, dated April 19th, 2020, was the Lee list, with links not just to LBCA, but to all of the lobular organizations and funds and consortiums. She was inclusive. She died 2 months later, 2 months that she apparently spent setting up an endowed biobank with BCRF. So talk about focus. Talk about taking the long view. It's hard not to be bitter, not to feel better, to feel that Lee got short shrift with that second primary diagnosis, metastatic ovarian, another understudied cancer. No, actually, Diane, she would say it's fallopian tube cancer, because she was always trying to advocate and convey a little bit more understanding and knowledge, even at the end. And in the end, all the wonderful advances in science and medicine were not able to save our friend. But Lee was pragmatic and ever kind. She did not hold grudges, not even against cancer. She would point to some larger lesson, use it as an opportunity to advocate for better screening, better imaging, better care, better science. Keep your eye on the prize and do not get distracted. Just do your best to contribute something constructive to the world to try and make things better for others, she would say. She did say, just keep pedaling toward knowledge and laughter and life. Just keep pedaling, keep moving forward, and don't forget to enjoy the ride. Thank you.
Well, that's a hard act to follow, Diane. Thank you. Such beautiful words. And thank you, Mary, for sharing Lee with us and having you here today. I can feel her with us today. So appreciate you making the trip. I am so just proud to be part of this Lob Mob Club. And just to give a little background, I was looking through old emails of how did I get involved? And because it's been so long, and I feel like as I've gotten older, my memory's gone a little. And I emailed Steffi after I think it was the 2016 first LBCA. And I remember emailing you saying, hi, I read about you. This is who I am. I live in Michigan. Can I send you my tissue? And like, I literally put that in an email, and she's like, who's this crazy lady? And she responded and connected me with Lori and Lee and said, we're starting the Lobular Breast Cancer Alliance. Let me put you in touch with them. And then Lee immediately emailed me, connected the three of us, and said, we're doing this white paper. Will you sign it? We're moving full steam ahead. And I was like, yes, I want to be part of this. I want in on everything, all things lobular. I was frustrated myself, being treated in Michigan in my other life, I work for a biotech company, so I'm very science-based, and I'm like, why isn't there anything on Lobular? What's going on? And I really felt compelled to help make a difference and help be part of this change. So I jumped in 2 feet, and Lee was such a mentor to me personally, was an educator. She was such a supporter. I mean, I can't even add on to all the things that has been already said to her, but to me, she really was a mentor, and she made me feel as if I was the most important person in the room or on the telephone or in Zooms where we spoke a lot. And she really propelled me to start a foundation and helped me not be afraid and use my voice. So I started an event back in Michigan, Ann Arbor. It's called Uncork for a Cure. It's a big wine food event. And I started in 2016. My physician is Dr. Dan Hayes. He could not be here today, but his colleagues, Jimmy Ray and Christine, who spoke, and others are all here, and Dr. Aaron Cobain, all supporting and representing. And after a couple of years of doing this event, I started talking more with Steffi and Lee and Lori, and I said, we need to study lobular breast cancer at University of Michigan. So I was like terrified. I'm like, oh my God, Dr. Dan Hayes is a gentle giant in the field of breast cancer. I asked him to have lunch and sit down, and I looked at him and I said, I want you to study lobular. And he like looked at me, and I don't know if he was like, are you serious? And he said, okay. And because I raised a bunch of money, and I gave it to University of Michigan, but I was very direct, and I said we needed to do this. And that was because of Lee pushing me. She was like, don't be afraid. It doesn't matter. They'll listen to you. And I'm like, okay. And so I was very excited that he really did listen to me. And then I pushed myself and started my own 501c3 and established a foundation, and we just started growing year after year. So I'm the founder of the foundation. I don't like talking about myself. On the patient advisory board at University of Michigan, and one of the founding patient advocates for the LBCA. And because of Lee, I truly believe that she really did spark a movement in me. She made me not be afraid just because I'm talking to a world famous oncologist, a researcher or scientist, that I had a voice and I mattered. And just because I'm a patient, I mattered. And so we established the foundation in 2019. And I'm proud to say that over 7 years, we've raised over $2 million. The funding, and I know Lee would be proud, we have funded so far over 12 research projects on invasive lobular breast cancer. A few of them were presented this morning, and some were in posters. So I felt it was very important to me. And when I spoke with Dr. Hayes, I said, you can't do this alone. I know you're like the person, but this is all based on a collaborative effort. And I felt very strongly after meeting Steffi and Adrian and coming here in 2018 and having dinner and visiting their lab. And I said, I want you to be part of this. Like, you're it with lobular. And Dan was like, okay, we can do that. And so we established some collaborative projects. And then we have expanded with Matt Sikora and University of Colorado and Karmanos Cancer Institute. And I really, truly believe that the main focus of the foundation is funding research, but it's putting the right people together to have a collaborative effort, to put your egos aside, share your data, and move forward, because that is the only way that research is going to be moved. And regarding Lee, and she left a fund to the foundation last year after she passed, and I was just floored by it. I was so moved by it. But I felt very strongly that we needed to carry her legacy in a very meaningful way. And what better way to do this was to establish the Lee Pate Memorial Lectureship. which really was going to honor her memory by ensuring her legacy would be illuminated with progress and hope, and that education and research, we could really change the course of lobular breast cancer and the research moving forward. So I'm proud. I'm proud to be in this room to play just a tiny role. But to all the advocates out there who are here, the patients who may be new, if I can leave you with one pearl, is don't be afraid to use your voice. And to the physicians and scientists and clinicians, thank you for listening to us, because, you know, we're smart. We read up, we want to know, we want to have a seat at the table. And I feel like Lee really made that happen. And she laid the groundwork for all of us. And now it's our mission and our job to really propel this and carry it forward. So thank you. And I just am proud to be here. So thank you.
I don't quite know how to follow this. You know, I'm hearing millions of dollars raised, you know, seats at some of the most important tables in the US. And it's a pleasure to have you, Lee's mum, sitting in front of me. I know that Lee visited Ireland shortly after she did a bucket trip with you. I read about it on her blog. She just headed off to Ireland 2 days later. So, you know, she did lots of crazy stuff. And I didn't know her. If I'd known her when she was in Ireland, I certainly would have met her. But I never did meet Lee. So, you know, we planned to meet at the 2020 Pittsburgh conference, and COVID got in the way. Had other ideas. We planned to meet in Utrecht. We didn't meet in Utrecht. But, you know, I spent hours of screen time, shared screen time with her. She helped so many of us here, but so many of us who are not here, to make sense of a diagnosis that, when we get it, makes no sense to us, because many of us, you know, do our mammograms, do our checks, we're health conscious, we're into our women, and then you get this diagnosis that just floors you and, you know, sweeps the ground from under you. And I think just the glob mob, it's so apt, because when I read Lee's the paper, not the paper, the article. Thank you, Diane. The article. It was one of the first things I read, and I'm always sharing it. I share it with everybody who's newly diagnosed, because it just gives such a clear understanding of how you feel as a patient when you're diagnosed with globular. And it's globular. That's what she heard. Globular. She heard globular. What is this globular thing? So I think to have us now called the glob mob is, I think she'd have a big smile on her face at that one. I want to thank Steffi and the committee just for having us here and to be part of, somebody said earlier, such a lovely table. I love quotes, and one that reminds me, and I think Lee you know, epitomizes that. Never doubt that a small group of thoughtful, committed citizens can change the world. Indeed, it's the only thing that ever has. So that's Margaret Mead quote. She was a US anthropologist. And when Steffi asked me would I like to come and be part of this? Of course, yes. And then what was I going to call it? I said, sowing the seeds of change. It was immediate, because, and I'm sorry if I'm repeating some of the things that have already been said, but when you haven't met this dynamo, there's only so much information you can, you know, get your hands on. And so I think we're all sharing little bits of what impressed us most about her. So before she was diagnosed, Lee cycled across America. And these words I love, because, you know, I often think, where do we as patient advocates get our, you know, strength and determination to carry on? Because you do get knocked back. You get knocked back all the time. You know, you make lovely collaborations with researchers, and then there's no funding and the researcher's gone. And, you know, it's a lot of starting over, and a lot of effort goes into doing, you know, to making those small baby steps. And that's where we're at in Europe. We're by no means where you guys are at here, But it didn't matter to Lee because Lee just wanted us to succeed. And she was so inclusive, and she talked about international collaboration. And that's why we're all here. So wait till I see, where am I? I'm lost. You know, we often talk about the E-cadherin and the loss of E-cadherin and Lobular, but she was that E-cadherin. She was the glue that, but you know, she brought us all together. So you know, she certainly is the glue that has inspired us to really bond as advocates in Europe. And she chaired meetings, you know, all through COVID. She met with us on such a regular basis, preparing us for coming here, preparing us for, you know, presenting a poster, our first poster here. And thankfully we had Laurie then, who was such a dear friend to Lee, and we're just incredibly lucky. So how we honor Lee in Europe, and how we hope to continue to honor her. So like I said, she celebrated our small achievements. We now have 10 patient advocates across Europe. We're building on it. We've got a patient leaflet to help with education of Lobular. It's now translated into 21 languages, with 2 more coming on board. I think we met somebody here that's going to translate for us. So, and there's no money involved, by the way. If you offer to translate, it's time. We're looking for your time, and we'd be delighted with it. So, yeah. So, honoring Lee's legacy, it's just so important. Tona from Norway, sitting in the audience, we came a couple of days early. We wanted to share some time together, but we wanted to be in Pittsburgh, where we had hoped to, well, certainly where I had hoped to meet Lee. I'm going to be a little bit longer than I should, but we were out walking in the park. We were coming back. We'd had lunch. We'd done the Mount Washington. We talked about her. Oh, not all the time, but we kept coming back to talking about Lee. Everything kept coming back to Lee. And we were standing in the park, and this huge butterfly, honestly, I'm not kidding. I've never seen a butterfly this big, but a big orange, orange-red butterfly floated past, and it fluttered around us. And the two of us just stopped dead in our tracks, right? And I said, that's Lee. We just stood looking at it, and it hovered for ages and ages and ages. So I don't know how many days I've been here. I've been here since Monday. I've only seen one butterfly. So, okay, I don't know how many people believe in, but I believe she's here with us. I believe her spirit is with us. I believe she lives through us all. And I believe that I won't ever forget her. I really regret not having met her. I really, really regret not having met her. But anyway, we don't deal with regrets. We'll never forget you, Lee, and the Lob Mob and what you stood for. And like you, we'll take those seeds of change, and we're going to plant them right across Europe. And we need your help, guys, to do it. And everybody in the audience, anybody who's willing to help us, will take the help. But I think the most important thing is, for me personally, and others that I've talked to, is I'll never forget the way that Lee made me feel. So I'm going to leave you with that.
I've heard Lee being called too many names, but never E-cadherin.
Can everyone hear? Yeah. I haven't actually got any slides, because I was like, unfortunately, I'm one of the people who never got to meet Lee. So I don't have pictures of us together and smiling at conferences. And then I thought, you know what? The best image for the people here today is me standing here, because without Lee, I wouldn't have been here today at all. I might have been here in a few years' time. I'm sure I've done lots of things wrong, but it definitely would have taken a lot longer. So I met Lee and Laurie on a Zoom call on the 24th of April, 2019. And I learned from Laurie, actually, whilst we've been here, they had a bit of a tag team. When Lee got an email, and she would send it to Laurie and go, can you check this person out, that they're legit? And I clearly made it through the checking process. And I got to have a meeting with them both on Zoom to talk about what I wanted to do. And part of the thing was I didn't know what I wanted to do. And kind of in the words of Lee, I just wanted to kind of lean in, extend a hand, change the sector. But I didn't know what that meant. I was kind of reeling from my own diagnosis, feeling incredibly alone in the UK because I couldn't find any information. I couldn't find people to speak with. And so firstly, meeting two other women who'd had lobular, which was the first time, was incredible. But then the support I got from them both. And the only other thing that I knew I wanted to do, because I come from a charity background, was I wanted to raise some money. So I said, look, can I stupidly run my first ever 10K? I've never done it since, for LBCA. And Lee's utter generosity and the sharing within the nonprofit sector, that when I ran it, she said, don't give it to LBCA, keep it there in the UK so you can get your brand and you can set up your website. And today I am very on brand. Those who know me, I'm rubbish at dressing. So one unexpected benefit of meeting Lee is she's given me a brand that I've learned how to dress myself with. So Lee, after the initial, Lee became my mentor, and I cannot express how important that was. I didn't know what a patient advocate was. I didn't know what I was meant to do. She taught me how to be a patient advocate, and not just an average patient advocate, a really good patient advocate, I hope. Whilst I'd run charities, I'd never worked in the health sector. And so she supported me in thinking of a structure for LBC UK. She taught me through the importance of having a scientific and medical advisory group. And so we set LBC UK up with that knowledge that Lee passed on. She connected me with researchers and clinicians, and I met Steffi and also Ali Sawyer, who's here at the conference, at a conference in Birmingham, because she'd done that thing that Diane mentioned of going, right, you need to write to these people before you get to the conference and ask them for a meeting. And from that, we really quickly established a really reputable scientific and medical advisory group for LBC UK. We're only 2 years old. We had our second birthday on Thursday, and Val Brunton's here as well, who spoke yesterday, is also on our advisory group. She regularly sent me resources, and I loved the way she'd do it, because she'd say, have you seen this? And I'd be like, oh no, I'll have a look at that. And then she'd say, have you thought about contacting this person? And I knew it wasn't a question. I knew it was a rhetorical question. And it was actually saying, you really should contact this person. She constantly challenged me as well. And she would get me to do, I remember doing a webinar with her with junior surgeons that I was like, it's a pan-European webinar that Christine Desmond was involved with as well. And I remember thinking, I can't do this alongside Lee, but I didn't really have any choice in the matter. I was doing it because she knew I needed to be pushed. And at Utrecht last year, when Lee was unable to come, she asked me to present alongside Steffi on the cancers paper. Which was utterly terrifying. I can't even say I stepped into Lee's shoes, but it was, she was absolutely determined, said, you can do this. And I think it's that confidence and support that she passed on to us. So I can't imagine the loss for Mary, for Anne, Lee's sister, for family and friends. I never had the fortune of meeting Lee, but I hope that from everything we've said today, from the work we're carrying on, and I think importantly, not just the work we're carrying on, but that we take Lee's values and the behaviours she demonstrated for us in that work. And also for me, I can't begin to tell you how many times in a month something comes up with the charity, and I just stop for a moment and say to myself, I wonder what Lee would do about this now. Thank you.
All right. It's an honor, to say the least, to be up here to talk to you about how Lee influenced my career and how she's continuing to influence the research in my group. So I did my postdoc here in Pittsburgh with Steffi and Adrian. I started my lobular journey in their group. And that was my first exposure to patient advocacy, to working with our local advocates in Pittsburgh. And at the time of the Lobular conference in 2016, I had been on my own in Colorado for, I think, less than 2 months. I was the babyest of baby professors when I met Lee. While Steffi has continued to be that bridge, as a Pittsburgh analogy, in my research and supporting moving forward, Lee is the bridge that helped me remain engaged in patient advocacy and learn how to communicate with the people that we serve. So Lee was not only important in those learning those tools, but really in giving me the confidence to be a scientist that played a role in patient advocacy and working with patients. So Lee and Lori reached out to me in early 2018 to play a role in the Lobular Breast Cancer Alliance, which at this point I'd been junior faculty for maybe 16 months. So it really meant the world that the people that I'm serving cared about what I had to say in the topic and wanted me involved in the program. I'll talk a little bit more about this, but Lee supported the establishment of my lab. Getting grants as an early career faculty is hard, but Lee was the advocate supporter for my American Cancer Society grants that kind of got my footing and continued to be for years. Lee helped me learn how to talk like a human. My wife jokes that my first language is science, not English. So when as our research was coming out and I would get emails and even voicemails from patients asking about anti-estrogen resistance and how our research implicated it, I would always not answer the call. But then I would email Lee and we would have a Zoom call where she would talk me through not saying anything completely stupid to a patient. And because of those conversations, that's what inspired us to put a lot more effort into communicating our science. I have a page on our website that was inspired by her where I tried to explain lobular and breast cancer research using only the 1,000 most common words in the English language. It's real hard. But it was great perspective that Lee talked me through and thinking about how we communicate and how a lot of what we say doesn't make sense to anybody but us. What was also really important that continued to carry through is that Lee genuinely wanted the patient and advocate community to know about what we were doing in Colorado and what we were building. When she became a Komen advocate in science, when kind of one of the first big lobular paper from my group came out, one of the first things she did was set up a Komen Advocates in Science coffee chat where we talked about the research in plain English over a webinar. And I did get to meet Lee in 2016, but that was about, unfortunately, as close as we got after that. There was a lot of Zoom, but this meant the world to us. Lee also was constantly pushing me to build more to support patient and advocates and connect them to the research we were doing. So this is just a snippet from an email in January that Tracy Cushing and I got from Lee, pointing us to a microgrant program through the BCCRF, Lori's group. And that was the square kick in the butt I needed to start putting together local advocacy events. And we pulled off our first one on June 1st. So, and Tracy talked a little bit about Front Range ILC yesterday, and it meant everything in retrospect, especially that Lee knew that we pulled this off with her encouragement. Finally, Lee believed in what we're doing in Colorado. And ask any scientist in the room, starting a lab is hard and getting back crappy grant reviews is hard. So it meant everything and more than getting grants that Lee believed in what we were doing. And there were more than once where she would tell me, this has to get funded, Matt. And I'm like, I know, I'm trying. But at one point, Lee and Lori conspired to help build long-term support for the lab. And then Lee and her estate left an extremely generous gift that is now established as the Lobular Breast Cancer Research Fund in Colorado. And I really hoped that this plaque would have been up so I could have gotten a picture with Lee before coming. But her name's going to be on the wall on campus soon. And it means everything that her name is attached to our program moving forward and all the research we'll be doing. So with that, I'm going to spend a few minutes talking about the project that I think Lee was most excited about and how we're moving it forward with her support. So one of the big interests in our lab is trying to understand how the estrogen receptor behaves differently in lobular cancer. And I could go on and on about why that's the case. But when I first got to the lab, we did a series of screenings where we wanted to identify what other proteins and molecules are partnering with estrogen receptor, which are physically working with ER inside lobular cells that isn't doing that in ductal cancer cells. So there's a whole bunch of screens, but the main thing is that the top hit after all of the screening we did was this protein called MDC1, and I'll talk more about this in a second. But MDC1 has been now a focus of my lab for several years in understanding why it works with estrogen receptor in lobular cells, but not in ductal cells. So I started writing grants about this, though. My first submissions were in 2018, and Lee and Lori were graciously my advocate partners through LBCA and talked me through the lay abstracts and how to communicate this work. Eventually, it got funded in 2020. This has led to a lot of the work in the lab that is ongoing now on the genomics of estrogen receptor and MDC1. The QR code there's for the relevant paper. That was the topic of our coffee talk, and we had a whole bunch of work in this space that has gotten funding since. But one of the things that came out from this study is what I think excited Lee the most. And it's linked to DNA repair dysfunction. I'll talk more about MDC1 in a second. But what we noticed is that if we exposed ductal cancer cells in culture to chemotherapy, they try to fix their DNA. It caused damage. They try to fix it. So each one of those little puncta, those little dots on the nucleus, can I point? Yeah. Each of those little they're called foci, is physically the DNA repair machinery getting to a break site to fix it. So they're getting onto a break site and they're fixing those breaks. What we noticed, though, was in lobular cancer cells is that they're not, at least the ones we have in culture, they're not really good at getting the machinery to the break site to deal with it. And we saw this in multiple models. But this kind of got us thinking about, is there actually something dysfunctional about DNA repair in lobular that we haven't thought about before? And there's actually evidence of this in TCGA. So the Cancer Genome Atlas Lobular Report identified 3 transcriptional subtypes of lobular cancer, one of which seemed to be the most aggressive. What was weird about it is that in the transcriptional data, this subtype is more the absence of the other 2 subtypes. But what's kind of hiding in the paper is that what's unique about this proliferative ILC is in this green group here, is this elevated DNA damage response signature. So my lab has been keying on trying to understand what does that signature mean and how can we interpret that to the actual basic biology. And we kind of have alluded to a lot already in the meeting that there are contexts like BRCA mutations where if you have a dysfunction in DNA repair, that's exploitable with therapy. So in that context, I wrote a Komen career catalyst application, and Lee, as a Komen advocate in science, was my partner on it. And she read the grant. And my first draft, I was kind of focused on the mechanism, this synthetic lethality of DNA damage. And I used more radiation because it was cleaner from actually doing the biology. And I included some PARP inhibitor stuff because it made sense from the therapy angle. Lee read it, and Lee never minced words with me. She didn't play with how she had opinions on my research. And it was, Matt, PARP inhibitors are what matter here. Radiation isn't going to solve any problems. You need to reconsider how you're writing this grant. So I did. I rewrote like whole aims to realizing that what I had written made sense scientifically, but wasn't going to move the needle for the grant. In that context, though, there are a lot of reasons I didn't focus on PARP inhibitors. We've talked a lot about already how uncommon BRCA1 and 2 mutations are in primary lobular cancer. Same with P53. In general, there's not a lot of evidence of scars of BRCA-ness. Demir showed some data from one particular signature, but in other data sets in general, lobular cancers just don't have that mark of having overtly dysfunctional DNA repair. And in our lab, we'd already kind of seen some dissatisfying or unsatisfying data with other classes of inhibitors. And there's weird stuff about growing lobular cells in culture versus how PARP inhibitors work that just make the experiments tough. So there are a lot of reasons that I hadn't focused on that. But Lee didn't care. She kept pointing out, like, this is what matters, Matt. I don't care about radiation. This is what matters. So that has been ringing in my head for a long time. So I mentioned MDC1 as the focus here, and I want to come back to this for a second because I think this is actually pointing to what maybe a gap, and I need to talk to Leonie and Demir about what they're seeing too now. So MDC1 is this protein here, and what it normally does is that upon DNA damage, which is a break in the double strands of DNA, there's a special mark in the histones that happens, and MDC1 binds it, and then acts as a scaffold, where it brings in the rest of the DNA repair machinery. So there's a whole class of proteins, including eventually BRCA1 and BRCA2, that come in response to MDC1 getting to DNA that initiate that repair. So we were curious because we identified MDC1 as partnering with ER and ILC. Is MDC1, is the proteins that it's work with? And in this universe down here, is it different in ILC? And this is work from Joe Sotnick in my lab. He did MDC1, he profiled all those proteins that were with it. This is kind of clustering or seeing similarity between different cell lines. And we did 2 lobular cells and 2 ductal cells. And what caught our attention immediately was that how MDC1, who it works with in lobular cells, looks kind of like one of the ductal models that's actually a BRCA2 mutant. So whatever MDC1 is doing in lobular cells looks more like a BRCA mutant than other IDC cells. And if we look at the classes of proteins that we identify, this is just its classification of the different proteins by their function. We do get a bunch of general DNA repair machinery in the ductals and lobulars. We do get other types of DNA repair in the lobulars that come down with MDC1 that partner with it. But the big glaring absence is that homologous recombination machinery, where we only see that in the MCF7s. We don't see that in the lobular cells or the BRCA2 mutant ductal cells. If we zoom in on it, long story short, there's a whole class of proteins that don't seem to be showing up as partnering with MDC1 in these lobular cells, but it does in the repair competent ductal cells. So whereas we don't see those BRCA1 and BRCA2 mutants in lobular, is this kind of a alternative mechanism of BRCA-ness in lobular cells that we just hadn't appreciated until now. So this is the entire text of the last email I got from Lee. The Coleman, that grant I talked about didn't get funded, much to her chagrin. But the latest mechanism came out and she thought maybe I should try again. So we are trying, Lee. So my lab is really excited about trying to understand what this potential DNA repair dysfunction is. We want to identify how to better define which tumors these are and how to identify the signature. We want to understand what drives that, whether it's MDC1 or something else, and we want to exploit that dysfunction. So we have a couple posters. Most of my labs here today. Joe and Jordan presented yesterday, but I'd encourage you to bug them anyway to learn more about what we're doing in this space. And this is like the most unpublished of unpublished data. This just rolled off like a couple weeks ago, but we are trying PARP inhibitor talazoparib, which is approved for metastatic breast cancer in these lobular xenografts. These are just conventional MM134 xenografts. And what we're seeing already is talazoparib, the PARP inhibitor, is outperforming fulvestrant, which is great. It looks like they're still growing, but we still haven't even gotten the H&E back yet. But I can tell you these tumors are pretty messed up. There is something there's a unique kind of sensitivity to the PARP inhibitor that we're just beginning to scratch the surface on. So with that, I don't do any of the work. My lab's here. But again, this is my persistence in this and trying to get it funded is all because of Lee. And now, even now, the local and the rest of the support that Lee inspired from my other advocate partners, including Lori, Tracy, Donna Charlevoix, who wanted to be here but couldn't because her bone mets are so awful and inconvenient, as she says, Sue Corrals, and now the rest of Front Range ILC. Thank you for letting me talk today.
We're running a little bit behind, but we have one last presentation, and this is from Dr. Huda, and what a very important initiative. You already heard a few initiatives from Lee, and this is something where Lee worked with BCRF on, and Dr. Huda is going to explain it. Actually, I should say Dr. Huda, together with Flora, did the yoga teaching this morning, which was really, really nice. Thank you, Jack Moon, for that too.
Can you all hear me? Yeah. Perfect, thank you. So we have heard a lot in a day and a half about the unmet need of well-defined ILC models. And we had two great talks in the morning from Dr. Biskin and from Dr. Lee, talking about the models for ILC research. And it's not just the researchers who have identified this need for having good models to study the disease. It is also Lee, who also understood the importance of having good ILC models to study, to do the research. And that's why, among many other things which she did as a patient advocate, which we heard from the panelist over here, she also donated to BCRF, Breast Cancer Research Foundation. And we feel very fortunate to be part of that program that BCRF has chosen us, the Hillman Cancer Center, and the MSKCC to oversee the process to develop those ILC models for research. Good morning, I'm Jagmohan Huda. So in this last few minutes, I'm going to give you a snapshot of the process for developing this ILC legacy project that we have, which Lee has envisioned, and we are trying to make it a reality. So before going forward into the project itself, I would like to quickly introduce you to the team. At Hillman Cancer Center, we have Dr. Steffi Oosterijk and Dr. Adrian Lee, who are spearheading this initiative. We have Daniel Brown from Institute of Precision Medicine. He is there in the audience, who is a really, really critical part of the project. he takes the initiative of developing the organoids coming out of the patient tissue samples. And then at MSKCC, we have Dr. George Rees-Philo and Dr. Britta Wegelt. Now, let's define the project. Like, what are our aims? What are we trying to build here? So the overall project can be defined with just 4 aims. The first 2 aims, they really focus on obtaining the patient tissue sample, getting all the clinico-pathological data, and then developing an organoid from those patient tissue samples. The next 2 aims focus on characterizing those organoid models, because as we have heard, well-defined models is the unmet need to do ILC research. So we really heavily focus not only just generating the models, but also then characterizing those models. This slide gives an overall snapshot of what the project looked like. It starts by obtaining a patient tissue sample, which is then divided into 3 parts. One of them goes into the FFP, which is used for digital pathology and getting all the pathological information. Then, depending on the size of the tissue sample, one part goes to the IPM, where our organoid is created. And after the successful creation of the organoid, the patient tissue sample and the organoid, they are subjected to sequencing, single-cell sequencing and bulk sequencing as well. And once we collect all this data, along with the well-established organoid, it's all put into a repository. We call it as BCRF ILC Legacy Portal, where all the information about the organoid and the patient tissue sample will be deposited, and the researchers can go to that portal and identify the model of which could be relevant for their research. Now let's look a little bit more deeper into the process of developing an organoid. So we also, in the previous slide, we have seen we call them PDOs, which are patient-derived organoids. And the organoids which are developed through this program, specifically, we call them LEOs, Legacy ILC Organoids. So I'll take you through the journey of a patient tissue sample becoming a LEO. It's not easy. I can tell you it's not trivial. This process requires a lot of patience and hard work and dedication. So it starts by obtaining a patient tissue sample, which then goes to the IPM, where an organoid is generated and established. So that's a tedious process. And once that's complete, it goes to Leo Strike Lab here at Hillman Cancer Center in this building. Then we culture the organoid to look at its phenotype, whether it looks like ILC, which is a grape-like structure. We subject it to immunofluorescence, multiple rounds of immunofluorescence, to make sure it has a loss of ECAD and cytoplasmic P120. I know we have had talks where we have discussed about ECAD still remaining in the ILC and non-dysfunctional, but we primarily focus on the loss of ECAD and the cytoplasmic P120. Then we subject it to STR profiling to make sure it's authentic, that we haven't mixed anything. After that, we perform the characterization through genomic sequencing and then bulk it up to put into a biobank where it could be used for distribution to the researchers. We also perform the phenotypic assays, as we have heard in the previous talks, the importance of having ER-positive models for ILC research. So we'll perform phenotypic characterization, and then the journey ends. For a Leo organoid, it's in the biorepository. But the journey doesn't really end. For a Leo organoid, from there, the journey actually begins, because now it's available for researchers, for you to use it and do research. Now let's look at some example data which we have collected in the last 1 year and what kind of progress have we made so far. So this slide will give an overview of the study which we have performed on a patient who had bilateral bone metastasis. The idea was here to understand that once we develop an organoid, how close is it to the originating tissue sample? So we took both the patient tissue samples from the left and the right and developed organoids, again, respective organoids from the left tissue sample and the right tissue sample, and all 4 samples were subjected to single-cell RNA sequencing. Now, this middle portion could look a little bit complicated, but if you look at the middle portion of it, which says BOM epithelial, that's the bone metastasis tissue sample from both left and right. It shows genomic characterization, clustering. And we can just focus on the colors. And the left and the right are the respective patient-derived organoids. So what we see here is the heterogeneity which we identified in the patient tissue sample is maintained in the organoid. And as well as we can see in the graphs below, which are where we have quantified these clusters, we can see it very closely resembles the patient tissue sample. So overall, from this slide, we can say that the organoids closely represents or recapitulate the disease from the original tissue sample. Next, once the organoid is created, we wanted to understand that once we culture it into subsequent passages, would it change or would it still remain genomically stable? So for that, we took an organoid and we, subject to multiple passages. Like here in this example, we can say we started from passage 13, went all the way up to passage 31, and collected almost seven samples from within those passages, 13 to 31. The top panel shows a clustering of all these different passages. And what we can appreciate here is they're all mixed, which is nice. That's what we want to see, that they're all mixed. There are no batch effects. The bottom one is the gene expression clustering analysis. Here, what we see is that through different colors, that there are 7 different clusters of genes which have been identified within those different passages. Then we further quantified those. And as we would see on the x-axis are the different passages from passage 13 to passage 31. And what we observe is there are certain clusters which are more dominant in one passage than the other, and there are other clusters which are more or less are similar across the passages. So overall, from this slide and the previous one, we can say the organoids are really good models to study the ILC disease because they retain those features as well as they are genomically stable over multiple passages. Now, let's look at the progress that we have made. We have given some examples. We have talked about the overall project, but what have we achieved so far? We have had 21 attempts to make these LEOs. And as I said, this is not a trivial process. So far, we have 8 PDOs which have been established. Out of those, as we say, we do multiple rounds of immunofluorescence. So we have had 16 PDOs LEOs on which we have done these immunofluorescence. And out of those, 8 underwent 2 rounds of immunofluorescence, and only 2 PDOs undergone 3 rounds of immunofluorescence to confirm that they have a loss of ECAD and a cytoplasmic P120. In addition, we have 3 LEOs on which we have performed whole exome sequencing and RNA-seq. And as we speak, currently there are 2 organoids which are currently undergoing single cell sequencing. In addition to that, we had 2 samples which are completely digitally. We have performed digital pathology and we have obtained the data for those. So the work is in progress. We are getting there. We are building the repository. So this is a tremendous progress within 1 year. Final thoughts. I think from presentations which happened today morning and the data which we have so far, we can say that organoids could serve as really good models for ILC research, but they need to be well defined. That's why we are determined to continue Lee Pate's legacy, the vision she had of creating a biorepository of these well-defined organoids for ILC researchers, for you, for everyone, all of you who are here, to use them, to find effective treatment options and strategies for the patient who are suffering with this insidious disease. With that, I conclude. Thank you so much.
Thank you, Jack Mohan. Thanks to all the panel. And Mary, thank you again for everything. And yeah. Thanks, that completes this morning session.